Uh, Hello, everyone. So we will be shortly starting with the keynote session by Dr. Anil Rai. I request all of you to please kindly wait for 2-3 minutes more. Thank you.
Good afternoon, everyone. I'm audible?
Yes.
Yeah. Namaskar, madam. Namaskar,
Yes,
madam.
ma'am, you are audible.
Janak, madam.
Yes, I'm able to hear you and very happy that I got this brochure and I registered myself because of the subject is very close to my life.
Yeah. Yes, so Dr. Rai is there because we are already late. So,
Yes, ma'am, he's here with us.
so shall we start? Participants are going to join without waste of time. Let us start, Neha.
Yes, ma'am, I will just start. I think Dr. Karthikeyan is with not us. Okay, so I will initiate this.
Yeah, just wait for some time. So meanwhile, Dr. Mark, I would like to know about how you are in touch with Professor Stephen. Are you collaborating with Professor Stephen?
Well, we're co-workers, really. I mean, we work closely in the Department of Electronic Engineering on projects related to robotics, data analytics, sometimes with medical topics. Um, I suppose, because we're all part of the intelligent systems and microsystems group, really our focus is being able to gather data effectively, to be able to analyze the data, be able to make use of it from a machine learning perspective. And with respect to this workshop, of course, a lot of the data gathering of interest is field data. It's real data. In Steve's case, that would be data gathered from humans, but here we're looking more at data that's gathered from outdoor environments, from crops, from fields. And of course, the reason we want to do that is because you can get better repeatability, you can do better science, get better error bounds on your data if you're using robotics and autonomous systems. So, I guess my message, and probably Steve's message, would be that you're only as good as your data. So you do, echoing some of the sentiments at the end of the last session, you do need to focus on the quality of your data, the repeatability, and the automation by which you get that data.
Yeah. Thanks. So we'll go ahead collaborating, sir. I'm hoping that you can visit, we'll invite, in fact, in IIT Indore as well. Professor Stephen keeps on coming multiple times since 2015, but due to this COVID thing, probably in Delhi, one conference is coming. It's an international conference, RAND conference, and he will be visiting in November and December. So we expect that you can also visit with him, and we'll plan later a short visit to IIT Indore also.
We'd be happy. Please keep in touch.
Yeah. And thanks you are here with us and sparing time to listen, Dr. Roy. He will be giving a talk. Shortly, Dr. Karthikeyan will be with us and will introduce about the speaker. So can
Ma'am,
anyone?
he will take, he's just taking 2-3 minutes more to join in.
Okay.
I think he's confused in the link, so he will be joining shortly.
Yeah, that's the problem. That's why I alerted to Rajesh and everyone, because link is
Yeah.
different. That's the issue.
Yeah.
So Dipali, Deepika, please call other students to join. At least BTP students.
Yes, ma'am.
Jadeja, please call our BTP group.
Yes, ma'am.
Ma'am, we have Dr. Karthikeyan with us.
Yeah. So
Yeah.
let us start.
Yeah.
So
Sir, you
you're
are muted.
Oh, I'm really sorry, I got disconnected. So, can I start the introduction?
Yes, sir.
Okay, so I'd like to welcome Dr. Roy, Dr. Anil Roy, Assistant Director General of ICT, ICAR, has made significant contribution in the field of agriculture statistics, computer applications, genomic and bioinformatics, and econometrics, and policy research. And last more than 28 years, he is working in this field. His experience includes research, teaching, training in the field of agricultural statistics, computational biology, and bioinformatics, sample surveys and computer applications, modeling, and economics. He worked in the area of modern statistical techniques such as analysis of complex surveys, geoinformatics, spatial modeling, small area of estimation, ranked set sampling, variance estimation, analysis of large scale survey data, dimension reduction techniques, gene regulatory network modeling, gene expression analysis, protein structure prediction, phylogenetic analysis, microsatellite predictions, miRNA predictions, SNP analysis, cross variation. Can you hear me? I'm sorry. Okay. So, his most significant and appreciable landmark contribution ICAR having national impact is the establishment of Ashoka Hub for the Indian agriculture at Center for Agricultural Bioinformatics. This is CAB. ICAR Agriculture Statistics Research Institute, New Delhi. He is instrumental not only initiating disciplines in bioinformatics in ICAR, he is also professor in bioinformatics in PG school, IARI, and responsible for initiation of course in MSc and PhD programs. He taught several courses to students of three disciplines like agriculture statistics, bioinformatics, and computer applications. He guided more than 22 students as a research guide, and he published more than 200 papers in international journals. So with this, is a huge achievements here. So with this, I am inviting to present his work.
Thank you, Dr. Saab. Thank you very much. And before I share, actually, I am basically PhD in statistics. Then I was working in modeling and simulations, and then moved to the computer science, where I worked in the area of data mining and data warehousing, and then now I am heading the bioinformatics. So actually, I have no discipline. All the discipline wherever I find interesting work. So I'll just share my slides. Most of the things which we have discussed today morning, the same are there with a bit detail. I hope it is visible.
Yes, sir.
Yes, sir.
Okay. So I'll make it. Okay, so again, good morning, everyone. And then I'm just going to talk about what we have already discussed and debated in the morning. And I'm not going to talk about the Ashoka because that we have established in 2013. And there we have established a National Agricultural Bioinformatic Grid. That's how its name is, Ashoka. Where we have 2013 we actually made it operational with 9 supercomputing systems. So So from there, we learned a lot of lesson has been learned, and we started our research in the area of computational biology and bioinformatics in ICR. Not ICR, some people are already doing, but it was a consolidated effort to have a research in the field of agriculture and computational biology and bioinformatics. And since then, we have developed a lot of databases related to the genetic markers of the crop, which are important for the country. And we also developed a lot of algorithms and tools as per the requirement and as per the problem which has come to us from our field institutes like different ICR institutes. And a lot of tools and algorithm has been developed, and they are all available on the National Biocomputing Portal site, which is there, which is available, and you can access there and see. And every year almost we publish, we have actually 12 scientists working in this area. Now we have published around, every year we publish around 60 research articles in different areas in a very high impact factor journals. So we learned slowly. Basically 2010 we started working in this area, and when this center was formed, Center for Agriculture Bioinformatics, and then we established ourselves in this area because there is a shortage of manpower for this kind of work. So I'll start my presentation that how we are using our high performance computing system for agriculture research and development. Only some important aspects I'll have presented. I'm going to present in this slide. We are doing in different areas, but some major areas I'm trying to cover. So it's not moving. Okay. So when we talk about the need, what was the need? As we have already discussed in the morning, that this kind of computing system is required when we were not having Ashoka in ICR system. So our scientists used to access the gene bank data, and still they are doing it. Some of them they are doing it, and they get access to some tools, small tools like BLAST and other things to do the analysis of the data, and it was very difficult. And we took around 1.5 years to think about that what is to be done, because a lot of our data were going to the gene bank, and we were not having that kind of facility to do the analysis. So that's how we have developed this Ashoka, which is totally dedicated to computational biology and genomic research, or omic research, you can say. So when you look to the need, it is needed for the food and nutrition, and projected world population will be 8 billion by 2025. And green revolution was there, but we are waiting for the next green revolution to come in the country. And I hope these are the areas with the support of computing where that will be the next green revolution we are expecting. And you can see that the earlier green revolution impact was not uniform globally, and basically it was scattered in some pockets, even within India also. It scattered some pockets of the country. Of course, that was very much required. Then when we look to the biodiversity angle, slides are moving very slow. I'll just unshare and then again share, I think.
Yes, sir, that is fine.
Yeah, because moving very slow. It is visible? Okay.
Yes, it is visible.
Okay. So if you look to the opportunities, so Worldwide, we have a lot of biodiversity when we look at the crops. And but in India, that is, we are more diverse, and we have a lot of opportunities to cultivate different other crops what we are cultivating presently. And if you see overall, only 150 crop species are commercially grown out of 7,000 species what we have globally. And only 30 crops provide 70% of the world calorie. And the major contributions are from wheat, rice, maize, which provides overall 50% of the plant-derived calories to the population. So, we have a lot of crops which are underused. If we don't use that, at least traits we can use, and we can exploit this biodiversity through the intensive research in field of agriculture and using molecular biology and breeding techniques. So we have a lot of potential, basically, which I want to emphasize, to use this through this kind of research and through support of the computing facilities. These are the major area in the crop, what we have already discussed in the morning, like increasing water use efficiency, per drop more crop. That is a slogan by our honorable PM also, because that is very important. And there are four broad categories what people are using to use this water use efficiency, like controlling stomatal aperture, controlling root development of the crops, so which root zone from which zone of the soil they should take the water, controlling photoperiod, and controlling light capture. So these are the areas where we are trying hard. Different scientists in the agriculture are trying to increase the water use efficiency of the crop, which is very much important, because water will be a major problem, going to be a major challenge also. And in African countries, it is already there. Then biotic stress management, we already talked about the soil salinization. The more irrigation you give, the more salinization is there, in Punjab, Haryana, and other areas where this problem is very much coming up. And now, basically, we are not able to break the threshold of the productivity, which is there, which is already established. And instead, we are declining productivity. We see decline in production and productivity in many of the areas of this, because of the salinization problem. And due to the climate change, non-optimal weather temperature, non-optimal pH. So these are major areas where people are trying to focus, and they are trying to develop a smart crop and their varieties, and biotic stress and plant pathogen relationship also, a lot of work is going on. So basically, when Dr. Sartu already told in the morning that genotype and environment interaction is very important when you talk about the crop production, although it's also true for the animal and other livestock. But crop production, that is very important. So same genotype may not perform better uniformly, rather, across different environments. So that's how phenotypic and genotypic interactions are very important. And it's very complex also. You have so many factors working into this, which are working with the genotypic character or genetic makeup of a plant, along with the environment sector. So you have epigenomics, transcriptomics, and so many other things are also there. So basically, it's a complex phenomena what we have to understand before going to have any genetic modification or any varietal development program. So as I told you that there is for understanding the phenomics or phenotypic characters of the crops, we have a moderate infrastructure. I will not say huge infrastructure. That is Nanaji Deshmukh Plant Phenomics Center in Pusa, which has been inaugurated by Honorable PM, and where we have a large number of scanners and sensors, So we have a controlled environment also, that plant to camera environment. So where it's all automated basically. And natural field conditions also, we capture the data, like we have a moving platform. So same genotype we put in a, and same we put the field also. So you can see the difference in the performance with the control versus natural field conditions. And then we capture it from the drone. Drone also, we capture the same, scan the same plot from the drone, and then we try to see how the control environment and field condition, basically you have to develop a variety which should perform better in the field condition. So, and then try to build a model and relationship among these different areas, and then evaluate the genotype, which will be the best genotype or parental lines for the future crop varietal development. So basically, we are mainly working with wheat and rice because that for different crops, different you need to have a different algorithms to extract the phenotypic characters, and if you see the genotypes, different genotypes, or you can say variety is different actually. Different genotypes have a different angles of leaf and different types of structure of the plant. So your algorithm has to be very perfect to extract the different phenotypic characters from the plant of the different genotype. So in this facility, actually, if you see that we have large number of sensors, and this is non-invasive phenotypic, we call it, because we don't destroy anything to get the phenotypic characters. So we have thermal, and we have a chlorophyll fluorescence, then we have NIR ranges are there, RGB cameras are there, visuals are there, and then we have hyperspectral and images in different areas. So for the same plant with the different angles, we capture the images, and we try to develop algorithms to extract the different phenotypic features out of this plant at the different crop growth stages. Basically, this plant goes on a belt, and it goes to the inside of the different chambers, and where there are photographs, images are drawn, and then it come out automatically. And every 4 to 5 days, depending on the how many plants, how many genotype you are evaluating, the image of a plant is being captured. So like this, we developed some of the algorithm. We are still developing, and we have a lot of challenges also for development of the algorithm, and that should be faster. Basically, you have millions of images generated from experiments. And similarly, we do for the field also. We grow, like this is one scene. You can see drought control, and there is no applications of this water normal condition, and you can do the imaging, and you can see that this is NDVI image only. So you can see the variations. You can match the variation from this basically from the field condition, and then what images you have drawn, what indices you have created. You can see this, how they vary for the different treatment combinations, what we call them. So this is one example. Just I am showing that if you have 40 varieties, and every variety is replicated 3 times, and you have 2 treatment, drought and control. So you generate how many images? You can see you can have number of images, and every 4 days the images are being captured with the different angles. And this is just example: visual camera 4 angles, and IR camera 2 angles, like that, IR camera 2 angles, like that. Lot of images is being generated, and we have developed a whole software for doing this analysis. Basically, the data is being fetched from there to the system, and then we have pre-processing, we have imputation for missing data, we have a lot of kind of relationship analysis, outlier detection, then feature selection. So whole system we have designed. Of course, that may be only working for the rice and wheat we have tested, but we have a lot of AI and machine learning tools algorithm, and basically backend we have used the R software tools, and we have written a script, the whole system, and then finally up to the GWAS analysis we have developed the system for the data analysis, and that needs very high end computations basically. So we have used SVM, cluster analysis, principal component for dimension reductions, then estimation of genetic parameters, so on, QTL analysis. So this whole pipeline has been developed for this kind of system, and but still a lot of scope is there for the improvement of the same pipeline. Then coming to the Omics Science, basically when we talk about this Omics Science, so basically most of the data is coming from this genomics, transcriptomics, proteomics, metabolomics. Now people started working in India. A lot less data is coming basically in this area, but these three areas are most dominating. And then we have initiated in the last 5 years, we have initiated work on the metagenomics. Actually, we promoted our scientists to work in the metagenomics because this has the biggest role for management of the natural resource management base. Metatranscriptomics, only few data sets are there with us with the metaproteomics and metabolomics. Still, we have not done. We do not have much experience on this. So these are the areas where our scientists made themselves available. These tools and techniques, they started using it at our center, and a lot of work, a lot of paper has been published. You can see online. So these are there. The basic problem is here again, the integration of the data. If you look at the biological process, so you have a DNA, then RNA, and then protein structure, and so on. So data for the same trait and for the same environment, a sequence of data is being generated through this biological process. And to understand that, basically, we need to integrate the data set horizontally and vertically. These two levels of integrations are needed, basically. You look at the sequence, then you have the protein and 3D structure, then families, small molecules, pathway network. And finally, we have to go to system biology approach. But most of the work are in this middle area of this curve, where we are stuck up. And still a lot of challenges are there to work in this area. And so a lot of things has to be done, and a lot of computational requirement needs to have a support for this, and a lot of algorithm has to be developed. When I started this basically work, so we thought that some algorithm may be there for doing analysis. Parallelization of the algorithm is a main challenge. Algorithms are there, but when you run on a HPC system, so within a same block, parallelizations are there of the sum of the algorithm, but across different servers, parallelization, I don't find any algorithms which can parallelize the whole system, which are there for the computational biology and bioinformatics work. So the present status, as we have already discussed, a lot of flood of data is there. Problem of storage. So phase one was the problem of creation and maintenance of the databases, and now shifting the analysis toward the large data set. Large data set means integrating the data set, which has the different kind of data set have different kind of errors. So you have to take care of everything and integrate it together horizontally as well as vertically. So that is very important, and metadata analysis still, because you can conduct one experiment and like salinity of rice. Suppose you conduct one experiment, but your experiment is a drop in the ocean. So large number of experiments are being conducted, and how you can synthesize and integrate together. This is a horizontal integration, and vertical means genomics, proteomics, and so on, metabolomics, everything together. How do you understand the whole system for development of crop variety or breed in case of animals? So integration of data and the linkage of data is still challenge, and of course, a lot of work has been done, but still a lot of work needs to be done. And the greatest challenge, as I mentioned, makes sense from the wealth of the data which is being produced. Actually, we tried to implement the big data platform, Hadoop MapReduce functions into this also. And still we are running a project in this area, but still we are not able to make it much faster as it was required. Some algorithm we have developed which made it little faster, but not the whole potential has been exploited from there. So we are working in this area. So when you look to the genomic data, how the genomic data and analysis of this genomic data has been progressed over a period of time. So old colleagues, if you talk about, so 90s, 1920s and 30s, we were talking about the population genetics. Then the DNA structure has been discovered by Watson and Crick. And then we have shifted the analysis. 70s, we had a lot of books on the advanced in quantitative analysis. And I also personally studied this area when I did PhD in statistics. So quantitative analysis books were there. Then Human Genome Project was launched. Then 90s, we started talking about the QTL analysis. So depending on the availability of genomic data and sequences, the whole environment keep on changing. And we talked about the marker-assisted selections, MAS, where some of the markers we identified, because getting a whole genome data was distance dream basically. And then 2001 genomic selection has been proposed, and this is how we moved forward. And then we get the genome from different animals and different cattle, dogs, and 2008 we talk about the bovine chip, SMP chip, genetic chip. So how you can check the genotypes, known genes only for known gene markers. Then you get the pig chip, and so many. Now we are have a series of. Then 2013, we got a faster technology, genotype by sequencing. And then this particular program, FANG, which is a European consortium programs, which make the whole genomic data much, much faster, availability of genomic data much, much faster. So this is how we move. So as I was telling, in the genomic selection area, which India needs to first step in agriculture, we need to move for the genomic selection. And it is much more than the mass. Mass is basically marker-assisted selection, where few markers are being identified and for which the varieties has been developed. But this genomic selection, where you incorporate all the marker information, so you can imagine how many markers are there, and then how do you incorporate this, and what kind of model you should have, and how what kind of training data set you should have. And then phenotypes. How can you use the phenotype? So it's a little complex process, but a lot of work has been done. European countries, China, and US, basically, Japan also. So this is how the difference you can see here. In mass, we use a few markers from the whole genome, but here we take all the genome and together. So, size of the agricultural genomes are very, very big, basically. Earlier, I thought that the human genome is very complex, but when we look to the agriculture sector and plant genomes, so they are much, much complex than the human genome. So now human genome looks to be very simple and very, of course, a lot of work has been done on this area also. You can imagine the complexity of wheat genome, which is hexaploid, and the whole world was behind that getting the genome, and they could able to do with some errors by 2014 only. Rice genome was very simple, so that was the first model plant genome of any crop. So basically, when you took the genomic selection, so you should have a training population, you have a genotype data, you have a phenotype data, you have to train models, that is AI, machine learning, deep learning, whatever model you apply, and mostly non-linear models are more successful into this area, but even simpler model may also work for some of the traits. So, and then you have to calculate the genomic estimated breeding value, and then make the selection of the parents basically for the next generation offspring. So this is the whole scenario, how we can do it basically. So now you can see that genetic gain, which we were talking, basically breeding cycle you reduce, especially cattle and buffalo, can reduce the breeding cycle from the 6 year or 7 year to the 2 years to 3 years. So that's how the genetic gain becomes very high. And you can see this graph where this is already plotted. The progeny testing scheme, if you go to traditional breeding, you go like this, and red color says the genomic selection. And these are examples where the people have achieved a lot of gain, genetic gain. But in India, somehow we miss this opportunity, and we need to do it. The only thing is that systematic phenotypic and genotypic data has to be there for the animals for different traits, what we are trying to do. And we are trying to push this in ICR system, and to some extent now we are start getting the data, customize our models. So the challenge is basically, as I told you, the basic challenge is that availability of the phenotypic data for our own breeds and varieties, which needs to have a systematically recorded along with the genotypic data. High dimensionality of the data, how do you reduce this? Multicollinearity is a problem because markers are dependent on each other. So how to segregate these markers? Other computational problem is large p small n problem. Like sample sizes are very large because very small, sorry, and factors affecting this traits are very large. So it's a non-statistical problem basically, how to do it, how to handle this non-normality of data, non-linearity of data. So these are some of the challenges in the genomic selection, but still there's a lot of scope. Then we talk about the metagenomics also in the morning. And basically metagenomics is one area which is now becoming part of the genomic selection also. In the model, we want to include the metagenomics also because, metagenomics give you the natural environment of the ecosystem. Basically, that is the ecosystem where the plant grows, whether it is soil or water, or so that is a very important criteria which has been realized later on. And now a lot of data we are getting from the field to get this metagenomics, and it's a human also. This is very important because your gut microbial community shows how healthy you are, basically in different performance, different traits. So same thing happens to the animals, and same thing happens to the plant also. So now we are working on the different projects on metagenomics, and basically one project we are running in ICR to microbial biodiversity of the rivers of ecosystem for health monitoring of the rivers. And Ganga Yamuna rivers also, we have taken the samples from different parts of the rivers, starting from where it is originating and going through this river cycle and taking the samples from the water sample also, and as well as the sediments also of the rivers. And then we try to study the aquatic system also into that water, especially the fishes. And basically, we have a lot of scope of discovering the remediating bacteria and probiotics from these kind of systems. And the impact of the human health on when they eat the fishes which are there from the polluting sites, and what kind of fishes are there also. That is so a lot of biomarkers you can discover from here, and a lot of very interesting results has came, and we published around more than 10 papers in this area from this project. Then soil resources also, we are trying to get the soil samples. This microbial population is highly varied depending on the environment. Like you take the sample in the morning, you take the sample in the evening. So there will be a difference. You take the sample during summer, during rainy. From the same soil, you get different samples. And from the different crop also, the microbial population also have a different community. That is a community study, basically, microbial community study. So it's a big area to explore, and basically that will means we try to how do you promote the growth promoting microorganism to the system. Similarly, for the gut microbe also, for animal gut also, we are taking sample. So it has a lot of potential for getting the bio remediation potentials are there, like in contaminated environment. How this, which are the microbes we can reduce the contamination in the environment. Ganga River, we find a lot of bacteriophages. There are microbial contents which can reduce the pollution of the Ganga. Similarly, metabolic potential of the soil microbiome, plant growth promoting microorganism, and new genes for the better agro products. So a lot of scopes are there. So we are taking this area also. And again, as I told you, it's not very simple because, large volume of data you need to handle, and cleaning of the genomic data, because you take the sample from the natural environment. So a lot of contaminations are there for selected genome. Because, so many genomes are there, they are in their sequences are interlinked. So how to assemble the genome, annotate the genome, and then community functions, how do you define? As I told in the morning that the genomes, to isolate the microbe, their function will be totally different than will be together with the other microbes. So this problem we have already discussed, so I'll not give much time to how to convert the C3 plant to C4 plant. It is the biggest challenge for India and China, and especially for the paddy cultivations, because we grow a lot of paddy, and the Western countries say that you are polluting environment. Your contribution to the pollution of the environment is more. But that is our staple food. So only solution is to convert this through some genetic alterations. Abiotic stress management, that is already being discussed, and this is how this whole cycle goes, and we need to have a network construction and data interpretation from the different angles, and then how we can manage this. Smart agriculture, finally, where now we are heading, and Industry 4.0, India needs to lead this area, and basically now our government target is to making India to make a hub of this Industry 4.0 Made India program. So basically now our target is to double the farmers' income on sustainable basis. That's how this workshop is there. And but the problem is that we have a smallholder, widely varying production conditions, dynamic market systems. So these are the basic problems which we are struggling, and then how to go for the smart agriculture under this kind of situation. Some of our panelists also discussed in the morning. So far, what we have done in agriculture is that we have used more input to get the more output. But the rising of input cost and resource constraints and other things are also there, apart from the pollution and contamination of the natural environment. So now we need to have a more efficient use of input and intelligent production options and smart marketing system to achieve the goal what we are setting us, what we have set now. And only solution is to go for high-tech and digital agriculture, basically. So giving the emphasis on this area, so our Honorable PM already announced budget announcement. You might have seen that now we are promoting the Kisan drones and other digital and high-tech services to the farmers. And there are some areas where we started working, as I told you in the morning, that we have started a network project where we are trying to use intelligent IoT-enabled tools. Dr. Mehta's institute is one of the area where we are trying to develop the machines for this, and drone-based monitoring and operation, smart application of healthcare, smart feeding for the animal husbandry and fisheries sector. So some of the animal institutes, also animal husbandry institutes and fisheries institute also we have taken into this program for doing this, monitoring and management of the crop health and protective cultivation, some of the smart, what do you call, multi-layer agricultural system, means that's what called protected cultivation. We call it semi-urban horticulture systems we started developing in ICR. Just we are trying to develop a model which can sustain in the country. Smart storage and logistic and blockchain also we started one of the horticulture crops, basically. So a lot of work we have started into this area, and we hope that within 2 years, next 2 years, we get some of the outputs. And this is the NEPA, we call it that project name, and Network Project for Precision Agriculture. So problem is only when you go to the field conditions, even for the institute like PUSA, which is in the Delhi, center of Delhi, transmission of data from the field to the servers, and then automation of the analysis, and then going back for the implementation, decision-making process, which has to be automated. So we have IoT devices, we have drones, we have other sensing devices into the field, but they have a different solutions. They have a different softwares where they are integrated. So how to join together and make a single decision-making platform is a challenge. We are working on that. And basically, these are intended outcomes from this project. So we have the crop survey assessment and advisory system we are trying to develop, monitoring and planning, traceability, soil health, smart farms. Whole operation has to be automated, pest surveillance, and yield assessment and monitoring, and better livestock and fish health produce. This is another program. This is the last slide. I think running out of time. Okay, we have 2 minutes. So now this is the one program which we have launched now recently, 2021. It is basically the Kisan Sarthi program which has been launched, and it is a whole digital platform, and we are trying to promote this whole digital platform, single platform for the farmers for different kind of services. And some of the services will be extended from the network program, different network programs which are running from the ICR. So here the farmer, we have a network of 731 Krishi Vigyan Kendras across the country. And we have linked all the Krishi Vigyan Kendras to this whole digital platform. And now if any farmers dial to the system a toll-free number, which is given here, they get directly connected to the Krishi Vigyan Kendra expert, and he need to register. If he registered, so we know that from which state he belongs. And BOT circles also we have mapped. So a lot of data is now coming, and this multimedia system is there. You can give and take advisory in any multimedia format, and all the advisory given to a farmer is recorded also. So farmer can also see what last advisory he got from our experts, and the whole telephone call is being recorded. And whenever next time he dials, he get the message. If you want to listen the previous advisory, so he can press 1 and he can again hear, because suppose sometimes farmer some advisory is given to the farmer, he may forget. So he can anytime he can dial the system and he can listen. So whole recording system and text messages and videos are likely to come on the system. So now the next challenge is how to make the early warning system. That's what now we are planning to have the next phase of this program. So whole cloud infrastructure we have to use, because around our target is to all PM Kisan farmer has to come onto this platform. That is around 12.5 crores farmers are going to merge with this database, and then all advisory systems. So we want to develop a knowledge management system and alert system, early warning system from the field level, that suppose some of the questions is being asked by the farmers in particular district or particular block, you can go up to the village level in the system. So if number of queries are related to a particular disease, this means there are some problem is coming, and that automatic alert has to be there from the system to the different people who are responsible for handling those kind of situations. Similarly for weather advisories. So this whole proposed architecture, now we are going to work with the Abhishek Singh ji is not there, but he is leading this program from the Digital India Corporation. And ICR and Digital India Corporation signed a MOU for working out on this program. And a lot of features. So all Kisan Call Center, earlier Kisan Call Center also is going to be merged with this program. And this will be a major program for the advisory system to the farmers of different kinds of advisories. And a lot of data is going to be generated, and then how to handle this data. That big data platform has to be used for this, I think. But still we have to try how to do it. So this is one of the challenge we are facing. Some other applications I will just go through slowly, and in time is over. So I'll end this. Thank you all for your listening. If you have any queries, then please ready to answer. Thank you. And thank you for the organizers for inviting me for this talk and giving me the platform to share what we are doing broadly, basically, not in details.
Thank you very much, Dr. Rai. Was excellent information regarding application of genomics in all the areas and precision agriculture. Here, I would like to also welcome a guest with us, Mrs. Janak Palta McGilligan. She has been honored Padma Shri for sustainable development and very active in the field of agriculture. Madam, we welcome you and would like to have your views.
Thank you. Thank you so much for this opportunity. And first of all, I appreciate that I have been almost, I should say, zero in this part of the learning about technology and how this will go. Is very useful for me to know where is India heading. But I have basic questions about, is this more pushed by genetically modified agencies? That's a question. Second thing is, how are we going to ensure better biodiversity, more biodiversity? Third is, looks like this will take us to more industrial food production rather than farm food production. And where is the human factor in the entire thing that will go? And I'm also very keen to know, because the status of agriculture colleges in India is miserable. The kind of this presentation I have seen, I want to know, is India ready with that kind of situation? How are we going to have smart and sustainable income of farmers? And the last thing I want to say is, humbly, I want to ask, it has been since 1972, I have been asking scientists that, because my first project in Indore was also with science and technology, that you're talking about advisory you will give. How will scientists learn from the farmers the indigenous knowledge and all the people who have been doing for centuries? And there are people who are doing organic farming and they're doing without any technical intervention. So these are few of my questions. I'll be happy to learn about it.
So, madam, I need to respond.
Yes.
Yeah. Okay. So, because so many questions were there, so I just missed few of them. So basically, the last question I start. Actually, the question you asked, that how scientists are going to learn from the farmers, because they are the people who are working in their field since long, means 50 years or 60 years or so on, same farmer. So that's how this Kisan Saathi program has been developed. Because earlier what we are doing is we were giving one way push advisory. We are pushing whether the farmer is growing a crop or not. He get the advisory from weather advisory and other advisory systems. So basically, our secretary has a presentation with Honorable PM, and then it has been advised that both way communication has to be there. So that's how the Krishi Vigyan Kendra is expert. And that's how we are recording everything. See, we have the farmer will ask the question, and the Krishi Vigyan Kendra people, they are not able to respond it, or they feel that is the area where we have to work on. Basically, this is the field problem. There may be solution, there may not be solution. So that will be taken up by the respective scientific organizations where it is coming. Second is, we have already compiled what you said, this ITK, we said, indigenous technical knowledge. That compendium is already there, where we try to capture the practices of the farmers in the fields. We are also, every year we have already a database where ITK we call, indigenous technical knowledge on the field. So where the current practices of the farmers which they are following. So that has been compiled all together. And then now we are looking for the scientific evidence to support that practices, whether it is okay or it's not okay. So that exercise also we are doing it. So this is how, and getting information from the field is very important for at least for the agriculture scientists. So that's how we try to communicate this two-way system now we made, because earlier you are right, it was only one-way system, and that's why we are trying to discard that and we are making it two-way. Next question, just if you
agriculture colleges, how are
situation? Okay, agree.
I have been watching all the only channel I watch every day is Kisan TV.
Okay.
And I listen to Kisan radio also. So I see that there is very poor learning aspect by the people who are talking to them. Number two, I don't see the people who are conducting those sessions capable of even having hands-on experience. So this is my concern, because I'm living in a village. I'm living on my own farm. I'm eating what I'm growing. I don't go out to buy anything. An organic, cow-based, simple farm. So I want to know if this is just a push from genetically modified. Is it a Monsanto's program? Are we going to be dependent on those countries for future? So is the technology bringing all those things and taking away biodiversity and cultural values of India? Those are my questions.
Okay, madam, actually you're right. When we try to domesticate and we go for a breeding plan, so basically what the genetic diversity we reduce basically, because we are trying to select the best parents out of the whole population, best parent in the certain trait. But for that only we have to preserve our diversity, genetic diversity. We have five institutions basically across the country, because one is to increase the production and productivity. That is one target. So when you go for that, so naturally we are going to reduce the biodiversity, because you are promoting best of the best. So like that, you are moving for the best of certain traits. So we have the National Bureau of Plant Genetic Resources, where all the collections of the diversity of different plants we keep, and that is kept for a very, very longer period. So breeders are using those for selecting a trait. Similarly, for animal, we have in Karnal. Similarly, for fish, we already have. And for microbes also, we have in Mao.
Yeah, excuse me. I have one question regarding animals. I was reading your thing about surveys and studies, and you're talking about beef and sheep. Why we need to do that in India? Why we are doing research on sheep and we are using that data? India is a Gau Mata ka desh or Dharti Mata ka desh, or why? Means I am a wife of a Northern Irish British. I have my in-laws there, but means I don't mind them eating beef, but I'm talking of India. Why we are studying beef and why are we studying sheep? Why we are not talking cow? Why we are not concerned about male cows getting deleted out of the system? I want to know those questions. I want to also know, is it Monsanto who is pushing this and we are following him?
Actually, madam, I am not the right person to answer,
Okay.
but I'll try to justify that. Beef, we are not talking. Beef, we are not doing research. Beef, we are not taking. Sheep, because some of the areas, our farmers, the very poor farmers are dependent on sheep in hilly regions. So sheep programs, to some extent, we have in Avikanagar is there in Rajasthan also.
Yeah, even buffalo is not part of your slide.
No, I have not taken anything. Cattle means buffalo is already included. Cow and buffalo is already part of the cattle. So buffalo is our major resources. Basically, our major resource is buffalo, and buffalo. Very few countries are working on the buffalo, and we have a different program for the buffalo. Basically, we have a dedicated institute in Hisar, and we have a dedicated institute in Modipuram. So that is a dedicated institute for buffalo. Cattle includes both buffalo and cow both. So that's how. So basically, genetically modified foods are not approved till now in India, and we are not doing anything in that area. We are so we are not talking about cross genomics. We are talking about the genomics only. So
Yeah.
still we
And
are
how are we going to make smart and sustainable? And what are we going to do with the agriculture colleges, which are most hopeless place in the country? Most useless institutions who have not contributed anything to the agronomy of the country or the food production of the country.
colleges, we agree, because colleges are suffering a lot. And basically, if colleges are affiliated with the university, agriculture university, we have 74 agriculture universities across the country. So agricultural university infrastructure is okay, but colleges, when you go to the college level, infrastructure is not there, and faculty is also not there. Faculty is also not there. So for that, actually, a big program, National Higher Education Program, started now, where we are trying to use the develop the course curricula. Course curricula, actually, we have make the same, but faculty is not there to teach.
Yeah, I have been a part of it. I have been invited to be included in the policies and all that at national and state level.
Right. So
very happy that something will happen, but I hope that all the good things and better things.
But in agriculture, madam, you'll appreciate that we have a common course curricula for all the agricultural colleges and all the universities. But the problem is that we do not have the faculty to teach that courses. We have a common examination system all across the country. All the admissions and everything is.
I get a lot of interns who need the certificate and come and they learn. And I had 8 girls for 3 weeks. They went back, and during the Viva, they invited me, and every girl said, we will go back and we'll have a biodiversity farm like here, half an acre land. We will go and work on the father's land. The dean asked me, none of the students throughout the history have come except want to get a certificate and they want to get a job. How did these girls come with the idea they want to go and work on farm? I told them, you are teaching here, there they are studying, आप पढ़ा रहे हैं, हम सिखा रहे हैं, वो करके देख रहे हैं, उनको बायोडाइवर्सिटी है. So I am really very concerned in related to that. Maybe you and other institutions here, maybe I should meet you some other time. I don't want to take your time, but I am very deeply concerned, and I'm saying these things especially to you and all the participants. Maybe some of you will take it forward, these messages and these concerns. An ordinary woman of 74 in India is concerned about India, what will happen to your children and your great grandchildren and grandchildren. I want your grandchildren. I was invited by parliamentary committee recently when I wrote a petition against the biodiversity bill. So I was very happy that they invited me, and this is what I said. Are we heading towards factory food? I want your children, grandchildren to get Indian desi food. I am eating sona moti in my house.
Oh.
And I am eating all the past varieties, and now I have been told in the market, I never buy seeds. I have been told in the market that जनक दीदी को जो गोभी के फूल की बीज चाहिए, वो अभी बाजार में आने बंद हो गए हैं. अब पैकेट मिलता है बड़े वाला और कंपनी का मिलेगा. So we will get everything from companies, we'll get everything from industry, we'll get everything from factory. So, but I have not, it will not bother me. I'm going up soon, but I'm concerned about your children and your grandchildren. What will happen to them?
Your
That's my
concern,
concern. Thank you so much.
right?
Thank you so much.
And
Thank you.
So we have to take care. Whatever you say, that is perfectly okay.
Thank you so much. And thank you, IIT, for having me.
Thank you.
Thanks a lot, madam, for joining and giving so much valuable inputs to us. The whole
Madam, Bharat Agri-Grid should solve our Janak madam's issues. We will put education as one of the component where the students, faculty can come and participate and address these concerns. How to keep a desi food from the Bharat grid. Maybe that could be one of the objectives should be. At the end of the day, we should get Bharat food, In addition to, okay, commercial, somebody want to make money, that is fine. But Bharat food should be available through our Bharat Grid initiative. Rai sir, I'm sorry I took 1 minute because I was so moved by madam's concern. And it is very, very important. We should address that. Okay,
Yes.
so thank you so much.
Thank you, sir.
Any other
Thank
point?
you, sir.
Okay, no, if there are no other points, so thank you very much to all of you for listening me. And some of
Thank you, sir. Your presentation was fantastic, very insightful, very futuristic, and very important and timely. So thank you, madam, for calling sir, and he is now authority to advise and guide us. He has a lot of experience. Thank you.
thank
It's
you,
from my
thank
side.
you, thank you,
Thank you
everyone.
very much, sir. It was very informative. And at Soybean Institute, we are looking forward to your collaboration and guidance from you.
Thank you very much.
Thank you very much, Dr. Roy. We'll invite you sometime in IIT Indore so that like IISR Indore and IIT Indore at one place and we can interact with you physically.
Okay,
Even our
Naren,
director
sure.
wanted this workshop in offline manner, but other guys said that we are most comfortable in online only. That's why it has happened like this.
Okay,
Thank you.
thank you very much. Thank you.
Thank you. Thank you very much.
Thank you.
Thank you, Dr. Mark, for joining. Dr. Mark is here, I guess.
I see. Ma'am, I have one question to Mark. So can I ask only 1 minute question before you close the session? Can you permit me only 1 minute?
Please go ahead, Dr. Khatri.
So, Mark, you have very good experience with robotics, IoT, genomics, and implementation at the field. What is your one-line recommendation for us? What we should take as a guideline based on your experience?
I can't speak very much for genomics specifically. In terms of analysis, data gathering, autonomous systems, my recommendation is to build systems that incorporate uncertainty and that are robust to missing data and changes in rule sets and in ontologies, I suppose. Don't expect your data to always fit the same model, and don't expect that the data is going to give you or is going to provide a consistent picture of what you're looking at. Try to be robust to that. And indeed, that's a very big focus of our research as well, is how to make autonomous systems robust to these kinds of challenges. And looking forward, I think, in terms of analytics and understanding these kinds of these areas, that's going to be a key challenge.
Excellent. Thank you so much. Ma'am, maybe you can proceed.
Yeah, thank you so much for joining, Dr. Mark. And I think Dr. Manisha Pathak is also there. Are you there? Manisha Pathak. So I think she's not, she is additional DCP in Indore in police department. So she was also interested in joining. Thanks joining by everyone. And the participants actually joined through YouTube. That's why people are not visible here. We shared a YouTube live streaming and most of the students are connected with that. So there is no question from that side. So now we can close this session. Thanks again for everyone.
Thank you,
Thank
madam.
you, Dr. Mark. I will be in touch with you. Okay.
Thank you, Dr. Irvina.
Thank you.
Thanks, sir.
Rajesh, you can play that video. Yes. Good afternoon, sir. Namaskar.
Namaskar. Good afternoon to one and all.
Thank you, sir, for joining and accepting our invitation.
Thank you, madam. In fact, today I am very much busy. I am not in my office. So I have taken the services of one of my dean. I am on the ground to Bhilwada. Bhilwada, we have got a college and Krishi Vigyan Kendra and all research center. So I have asked my dean to give me his office and stay outside of this room. I have got some important work.
Yeah.
Yes, sir. So I'm sorry, I have not prepared slides, but I will happy to speak on agriculture education and its different component which is visible in our present context and what will be its scenario in new education policy and how we will articulate the concept of digital technology, especially in just to increase our production, productivity and profitability at the farmer's end. So that will be my whole concentration.
Yeah, so nice of you.
Thank you.
So my colleague is Dr. Ratnapark. He is with us.
Hello, sir.
Yeah.
Just want to introduce him.
Sure,
He is a
thank you.
principal scientist in Indian Institute of Soybean Research Center, Indore. I told you during my visit.
Yeah. Yes. Welcome, Daksa. Very good. Thank you very much.
So it's nice to see you here in this workshop, and we are eager to listen to
Yes,
your
yes.
talk.
Yeah, so I am the session coordinator for Dr. Rathore. I want to introduce. Good afternoon, everyone. So we are back to this workshop after lunch. I am hoping all participants joined here. Dr. Neha, would you like to say something? I think she is not connected. So I am starting the session, otherwise we'll be late. Other speakers will be joining from abroad. So quickly want to introduce Dr. Naren Singh Rathore, Vice Chancellor, Maharana Pratap University of Agriculture and Technology, Udaipur. He did graduation and MTech from IIT Delhi in Agriculture Engineering and Energy Studies, respectively, and PhD in Farm Machinery and Power Engineering. He specialized in renewable energy technology. He beared various professional duties, including Director General Education and DDG, Agricultural Engineering ICAR. He is Dean College of Dairy and Food Science Technology. He worked as National Director in World Bank sponsored NAHEP during August 2017 to May 2019. He is recipient of 33 several awards and fellowships, including Young Scientist, Outstanding Book, Outstanding Services. He was the Chairman Steering Committee Consortium of E-Resources in Agriculture in ICR, and he published several papers and is a national and international person. So now, Dr. Rathore, please start your session. It's like 40 to 50 minute session and 10 minutes question answer.
Thank you very much, madam, Dr. Tiwari, and all other colleagues, those who have joined online. I am really happy to see this opportunity in this afternoon session. And although I'm sorry, I have not prepared anything. I am not even as per of your scheduled program and as per of the schedule, your point of discussion. I have not discussed anything so far because of my preoccupancy. But I'm sure I will justify my presence here and will try to speak some very valuable things as far as the agriculture education is concerned. Everybody knows that in 2016, our government of India has declared agriculture education as professional degree, just like engineering and technology and your medical education. So agriculture education earlier, before 2016, it was not a professional degree, although we were fulfilling all requirement, and fortunately we got it declared in 2016. because agriculture education is not only meant for human resource development, because generally what we are saying whenever we are talking about as far as the education is concerned, we used to say that education means creating awareness in the mindset of different masses as far as the subject matter is concerned. Even to certain extent, education gives an way out that how we can awaken the policy maker just to enhance our visibility as far as our subject matter is concerned. But apart from this, agriculture education has got a potential of a applied research and basic research. When we are talking about basic research means a knowledge generation, and applied research means a development of technology for improved seeds, fertilizers, implements, and insecticide and pesticide, whatever may be the component of agriculture. So essentially in agriculture education, we used to give major stress on development of applied technology as well. And it has got a hidden agenda as far as the technology outreach is concerned. So I can say that agriculture education has got a potential to expand our activity, not only in the field of teaching, but in research, extension, capacity building, training, consultancy, testing, certification, and technology outreach. And as such, agriculture education has got a potential to increase our production, productivity, and profitability. And that is the reason I remember in 2018 I have attended some APARI meeting in Taiwan, where I have represented our country. At that moment, I was really happy to see that a statement made by the many international agencies. They said it was agriculture education in our country, and thereby we could increase our production manifold. If you see in 1947, our production was hardly, as far the grain production is concerned, it was hardly of 49 million tons. It means a 4.9 crore ton. But now we have enhanced it to the 300 million tons. Almost you can say 6 times we have enhanced. So it is only because that we have introduced very systematically agriculture education in our perspectives. If we compare the neighboring country like Pakistan, Bangladesh, even Sri Lanka, if you see, Sri Lankan people have started number of reforms, but you can visualize what happened in last year, recent year. And it was only because they lost identity as far as the agriculture is concerned. Even there was some problem of a food availability. Whereas now in our country, we are not only concerned as far as the food security is concerned, but we can say with the help of agriculture, we reach to the position of as far as the your health security is concerned, as far as the immunity security is concerned. To a certain extent, everybody has realized that during the period of COVID, we have shown to the whole of the world. I remember one of my friend, he is a very big gun in a Pakistan Agriculture Research Institute. And what he said, Dr. Rathod, you will face his consequences because your population is more than 17% of global population. But I with proud, I can say it was our farmers. They have enhanced our production, productivity during this period. And that's why if you compare before COVID, our GDP contribution because of agriculture, it was hardly of 14.3%. But after COVID, we reached to the level of 19.9% GDP contribution. It is only because that we have got a very good setup as far as the agriculture production is concerned. We stand first position as far as the milk production is concerned. From 1999, we stand on first position. Last year, we have crossed 200 million tons of milk production. If we compare the even the our USA is producing 25% less than our own production. So that's why we can say that in our agriculture education, we show a different path as far as the how we can increase our production, quality, productivity, profitability, even a sustainability, even how to get rid of this climate change, how to federate the farmer into the business group, all unanswered questions one can easily give through the right component of agriculture education. And education, in fact, whenever we are seeing our whole production, we stand on second position as far as the fruits and vegetable is concerned. Even some material we stand on first, second, even few like 52% of mango worldwide we are producing. More than that of 26% of cashew nut we are producing. more than that of 67% of brinjal we are producing, more than that of a 37% of a cauliflower we are producing. So basically, if you compare and you see our overall production, one can easily say it is something because of a knowledge, because of a skill, because of a ability of farmers, and because of experience of a farmer, we reach to this position. So basically, our education is just like a root of a plant, and it is a hidden, it is not visible, but it is contributing, and it is most essential. Everything which is happening as far as the agriculture is concerned, it is something given by the agriculture education. In our country, we have got a more than that of 1000 university across our country and more than 42,000 colleges. But when we are talking about agriculture university and colleges, we have got only 74 agriculture university. Out of the 74, 64 is a state agriculture university. In all 28 state and 8 union territory, we have got some component of a agriculture university. Apart from this, we have got a 3 central agriculture university. One is in Impal, Central Agriculture University Impal, catering the need of a northwest India. Second is in Jhansi, central part of our country in MP. And third is a very important in our Samastipur, Bihar, Central Agriculture University in Bihar. And apart from this, we have got a 4 deemed university. One is a IERI, world biggest agriculture research institute, that is agriculture, Indian Agriculture Research Institute, New Delhi. We have got a NDRI, exclusively working for a dairy technology business. It is in a Karnal, that is you can say National Dairy Research Institute. Then we have got a IVRI, Indian Veterinary Research Institute in Ijat Nagar, Bareilly. And then very important component is CIFE in Mumbai, that is Central Institute of Fishery Education. So likewise, we have got a 4 deemed university, 3 central agriculture university, and 4 central university where there is a agriculture component is available, like your Banaras Hindu University, agriculture component is there. Muslim Aligarh Muslim University, that is also a component is available. So likewise, 4 central university having agriculture faculty, 3 central agriculture university, 4 deemed university, and remaining 64 is a central, our state agriculture university. And as such, if you see our classification, you will definitely appreciate that a 50 out of the 74 university, 50 universities are having a composite scenario, means they are teaching the agriculture along with the horticulture, fishery, and veterinary as well. And exclusively, we have got a 16 university dedicatedly working for a veterinary business. And then we have got a 6 horticulture university and 2 exclusive university of fishery. This is a second classification. So you can say that as far as our agriculture education is concerned, we basically having a very good setup. Out of 74 university, that is a government owned, public sector owned university. Apart from, we have got a, under this 74 university, we have got a 400 colleges, and out of these 400 colleges, more than 300 colleges are being accredited. But if you compare 1000 university of a overall university of our nation, out of this 1000 university, 600 universities are non-accredited. Out of 40,000 colleges in a general education, 17,000 colleges are non-accredited. Whereas in our case, out of 74 universities, 72 universities are accredited. And out of 400 colleges of agriculture college, more than 322 or 21 colleges are accredited. Apart from this, we have got almost thousands of private colleges. Nowadays, everybody is diverting their attention towards the agriculture business, and they are imparting a right education. So that's why we basically, if you see, as far as the agriculture is concerned, we are having a very good setup. But even then, this was my observation. I met with Dr. Kasura Ranjan, who was the chairman of New Education Policy. At the time, I was working as a Deputy Director General Agriculture Education. I got this opportunity to brief some of my views on agriculture education. But later on, I could realize that as far as our capacity is concerned, it is hardly 1% of the whole scenario. When I am talking that we have got a 1000 overall university, out of this 1000, only 74 is agriculture university. So less than that of a 1%. And same thing is true with our student intake as well. In a overall agriculture, in a accredited colleges, we have got only 40,000 or 50,000 student intake. And as far as the private colleges is concerned, around 1 lakh students are coming. So overall, if you compare the overall students in our university setup, you must be knowing that in our university setup, in a 1,000 university and 42,000 colleges, we have got a 3.96 crore student, just equal to the population of a Canada. Whereas in our agriculture setup, our capacity is small, and second, our quality, that basically was also a point of a concern. And our all honorable member of a new education policy, they suggested we will have to expand our quality as well. Quality means we should focus on how to increase the productivity, because this is general understanding. Farmers are under unrest. Whether you are staying in a Maharashtra or MP, or I am speaking from a Rajasthan, everywhere farmers are not happy because of number of a constraint. I don't want to go deeply in this case, because what is happening today, farmers are expecting something extra, and that is true because they are producing a lot of quantity. So that's why it is must that how we can fulfill their requirement as far as the net profitability is concerned. Government of India has targeted by 2022. We are supposed to enhance the farmer income to a double level. To certain extent, in some pocket, we have enhanced. What to talk about double, even triple, 4 times we have enhanced. But still, there's majority of farmers still they are complaining they could not reach to their income to a double level. So that's why there is a need that we are supposed to see that how they are fighting with a different type of problem. In agriculture education itself, it was suggested in new education policy that we are supposed to see and supposed to dispose of the problem which our farmers are facing. Because it is true, farmers are fighting with a number of problems. They have got a somewhat frustration as compared to service sector, because service sector has got a number of things which is in a quite appreciable stage. And even the most of the farmers are supposed to focus on quality as far as the production is concerned. Everybody used to say that a farmer should do extra work and they should develop their production. Likewise, this is a general thinking in most of the intellectual group people are thinking. But basically, we will have to see that if agriculture education has got some component which we can easily enhance our capability and our capacity as well and our quality as well, as far as the solving of the problem of farmer is concerned. That is a very much interesting thing, that agriculture education, if it is given a right attention, I'm sure we will be in position to fulfill. Because as I mentioned earlier as well, education, agriculture education, interestingly, it includes the component of capacity building, training, consultancy, testing, certification, technology outreach, along with the research, extension, and teaching, as of course. And essentially, because I was associated with the 5th Dean's recommendation as far as our syllabus is concerned, at the time we have discussed almost every dean of the whole of the country, and where we made a lot of deliberation. And essentially, I have concluded, my complete team has concluded that agriculture education has got a 4 important component, and we are fulfilling that requirement. When you compare these 4 components with the technical education and health education, in technical education, we have got more than 3,000 engineering college. And as far as their total intake is concerned, more than 14 lakhs students are getting admission. But in our agriculture education, hardly we have got a not more than that of 1.5 lakhs student, if you include private as well, private and public sector institute, not more than 1.5 lakhs students are getting admission. So basically, what we will have to see that how our agriculture education differ from this. Essentially, agriculture education, we used to give the whole technical support through the theoretical perception. Second is a practical exercise in every course, and it has been estimated that more than 67% of our syllabus is a practical oriented. And that is the reason why agriculture education was declared as a professional degree. I remember it was objected by UGC, and then we have calculated in details, and finally we have concluded that in our agriculture education, around 67% of our component is a practical. That is a second component of agriculture education. Third component, which definitely everybody, especially our all participant from the engineering background, they will appreciate that we have got a third very important component of experiential learning, hands-on training. And in fourth year, we used to give major stress. And because what is happening, we basically training them and we are imparting a right type of skill. I will discuss later on with that how 4T component we have arranged, means what is a student ready program. And fourth component which we used to focus, that is a leadership development. So essentially in agriculture education, which is these four component, all four component in integrated manner in a holistic way is not available in other. I also worked as a dean of engineering college, where I have seen that the old syllabus which was long back was reviewed and it is running. But fortunately AICT last year they have changed the whole syllabus. So I'm really happy to see that in engineering syllabus as well, new dimension has been introduced. But interestingly, in agriculture education, theoretical perception, practical approach, and experiential learning and leadership development, we used to impart in right way. And I'm always saying in a fifth dean's component in a report which we have submitted, it has got a very well, they had described that agriculture education include 1T, 2T, 3T, and 4T. I will explain what is 1T. 1T means a first only T means a temperate. When I am saying temperate means a balancing. This is the only education where we are keeping right balance between teaching, research, extension, capacity building, training, consultancy, testing, certification. Every component has got a some position as far as their contribution in overall perspective is concerned. That is a temperate. And second means a 2T. 2T means a even in a during the COVID period where most of the students were promoted based on their marks of a earlier subject, but we insisted because in our agriculture education, this 2T means a teaching and testing. These are the two essential components which we have introduced. So that's why in our agriculture education, when we are teaching something, when we are briefing them that what are their basics, fundamental theory and practices as far as the knowledge is concerned, and as far as the skill is concerned, when we are imparting experiential learning and hands-on training means how to do. So that's why we mentioned that when teaching is there, so testing, evaluation should be there. That is a 2T component that is essentially available in our course syllabus. And 3T means a timely action, trouble-free approach and transparency. Why it is so? Because in our agriculture, if you remember a quote given by our first Prime Minister, Sri Jawaharlal Nehru, he mentioned everything can wait, but agriculture can't. And this is true. Because what is happening, you are supposed to see when you are this time, every farmer is busy for preparation of Kharif. Kharif preparation is due because very soon when monsoon will come and when moisture will be available in our soil, we will have to grow something. And that's why timely action we will have to take. And the all like operation of unit operation as far as the plowing is concerned, land preparation is concerned, sowing, and then irrigation, then interculture operation, and then judging their maturity indices, looking that whether they are completely mature, then harvesting, post-harvesting, storage, processing, marketing. Every unit operation, basically a timely action is required. And even you are not supposed to make it cumbersome as far as the farmer is concerned. Nowadays, farmer has got a very good potential as far as the availability of machinery or mechanization is concerned. So basically, we will have to see that 3T component must be always available. We are supposed to have a timely action. Otherwise, if we will leave the right time, because you must be knowing in a Rabi season when we are interested to grow wheat, generally Government of India and Ministry of Agriculture and Farmer Welfare, they used to give a information and advisory to the farmers that before 24th November, you are supposed to grow. Because why it is so? Because we will have to see that what will be the total maturity time, and based on this one, harvesting should not be in high temperature. This year, unfortunately, during the harvesting of a wheat crop, our prevailing temperature was slightly more than that of a 1.7 degrees centigrade as compared to last year. And that is the reason why our overall production reduced by a 1.2 times. Because early estimate, we have concluded that we will cross 109 million ton as far as the wheat production is concerned. But unfortunately, we stuck to the 106 million ton. It was only because that during harvesting period, because of a high temperature, the production was slightly reduced. So that's why timely action is required. And 4T means a syllabus. Essentially, the syllabus we have designed. What I was talking about, fifth Dean's recommendation. We have taken this 4T arrangement. First T is a traditional, second T is a technology, third T is a talent, and fourth T is a trading. These are the four essential components. We are supposed to teach the student that traditionally how we grow. What was our traditional technology, indigenously and worldwide, and how things were available as at the time of independent and prior to independent, even during our Maharana Pratap regime, because everybody, I am from the land from where Maharana Pratap is a famous, and that's why we used to say that as far as the this trenching and terrace farming is concerned, Maharana Pratap gave this. As far as the water harvesting and conservation of water is concerned, during in 1536, that component was live. So that's why we will have to discuss something what traditionally it was available. And second thing, it means what is a technology which is available in present context and indigenously and at international level. And third very important component is that how we can increase the talent of student. Because what is happening every time, we are supposed to see that what is happening in present context, what are the frontier area of agriculture, and how our students are being trained in this frontier area so that they will become brand ambassador next year. And that is the reason in fourth year, a student ready program, which definitely you will appreciate. Ready means a READY. It is basically rural entrepreneurship awareness development yojana. Means in a complete fourth year, no coursework is available. It is expected from every agriculture college that they are not supposed to have a formal classes, but whatever things they have been trained during first year, second year, and third year, in fourth year they will have to apply exactly by their own hand in the field. And that's why this entrepreneurship scope has been added. That is a student ready program. And here it is expected not only a knowledge but skill. Knowledge gives an impression what to do, skill means how to do. And we are supposed to increase the ability of a student, means how to simplify and conveniently mold of the different on-farm application. And during this student ready program, even we are supposed to share our experiences. Sharing experiences means how to reduce the losses and increase the efficiency. And that's why 4T arrangement was systematically arranged. And for your information, under this student ready program, 5 different components we have floated. And these all 5 components is not supposed to undertake by each and every college, but out of these 5 component, at least 2 component one has to implement, so that students will be with a one complete year, they will be fully busy. Otherwise, what will happen? Student will roam here and there. And that's why these 5 component, very first component is a experiential learning. That is, you can say, earning while doing or while learning. That is a concept. Here, more than 416 experiential learning unit and type of industries the government of India has established in a different agriculture college. That is a strength of agriculture education division. And you will find everywhere, wherever you will go in agriculture, accredited agriculture college, you will find experiential learning unit. Like me say how to process tomato, not processing of tomato to the limit of 5 or 6 kg, but 1000 kg of tomato, how you can process, how you can prepare different type of organic manure. Likewise, there are number of things are there, and these are experiential learnings are available in every college, where student is supposed to undergo. He will purchase the raw material from the market, then he process, make a value addition, and finally keep all inventory, and then sell it into the market. And whatever profit they are generating, 50% of profit is being distributed among students. That is a really good component. That is called experiential learning. But many times what happen in number of university, especially finance department, they have objected that why we will distribute the money to amongst student, and that's why they have not accepted our proposal. Then we said instead of a running in a actual use, what you can do, you can have a hands-on training only. They say you establish the system, then make a repair, maintenance, and other activity and operational, and let us train the student without performing in actual use. They can understand how to dismantle the system, how to assemble again, what are the different repair and maintenance is available in the existing system, what are the different operational tricks are available. So likewise, it is a different type of hands-on training. That is a second component. Third component, which has got a very good visibility in number of agriculture colleges, it is that a industrial attachment in plant training. It may vary from a 4 month to 6 month, where students are going to any seed industries, implement industries, fertilizer industries, where they are undergoing their exact operation. It is expected that student is supposed to understand that how seed are to be processed, how a tractor is to be manufactured, what are the different unit operations that one can perform as far as the different farm implement is concerned. So that is a industry's attachment, and this is our observation, that most of the students are being absorbed there only after a 6 month period, because they used to create a very good impression to the management of that industries, and industries people are also thinking that we have already trained this student, why not to absorb them. So that is a basically reason why in our agriculture education we have got a very good status as far as the placement is concerned. You will surprise that more than 80% of the placement is there, and especially in agriculture. But when you talk to dairy technology, food technology, and even to biotechnology and agriculture engineering, more than 90% of placement is there. And that's why it is the only because of this industry's attachment. And fourth component is a RAWE. RAWE means, in short, if I describe this word RAWE, it is a rural agriculture work experience, where a student is supposed to see very closely what are the different problem our farmers are facing, and based on his knowledge up to third year, they will articulate their knowledge and try to solve this problem. And that is a basically purpose and basic objective of RAWE. So here students are visiting nearby farm along with a scientist of a Krishi Vigyan Kendra. You must be knowing our farmer science center. In every state agriculture university, this Krishi Vigyan Kendra is available, where a lot of scientists from a different disciplines are available. So along with these scientists, our students are going to village, getting their problem and trying to solve this problem based on their experience and based on their knowledge, based on their ability, based on their already experience. And they basically gives a right, and they are supposed to submit the report as well. And fifth component which we kept, that is a student project. Under this student project, few students, those who are eager for higher education, so they used to opt this student project, where basically student will have to identify some problem, and then he will have to do some exercise in the form of experimental setup and taking observation, whatever data they are collecting, then how to analyze the data, how to interpret the data, how to present this interpretation back to the original system, and then how to organize that system. These all things they are doing in the form of a dissertation. And then it is expected that same thing they will repeat in MSc and PhD. So at least during BSc program, they will be acquainted with what are the different steps are required in writing a thesis or a dissertation. So that is a fifth component, means experiential learning, hands-on training without business mode, industries attachment or in-plant training, and fourth one is a RAWE, and fifth one is a student project. So that's why if you see in a fourth year, our students they are being exposed with a number of a good thing, and ultimately sense of skilling and sense of trading, sense of a marketing is imparted in their mindset, and thereby the students will believe, and they will generate and they will gather their strength, and finally they will be in position to say that we will again start our entrepreneurs. I have seen many of our students, they have started their entrepreneur, they become an entrepreneur at the end of doing this one. And interesting thing is that excuse me, as far as the undergraduate courses is concerned in agriculture, we have got 11 well established undergraduate courses. Like engineering, means in engineering you have got civil, mechanical, electrical, mining, likewise. So in our agriculture we have got 11 disciplines. One is agriculture, Second is a horticulture. Third is a forestry. Then we have got a fishery. Then dairy technology, food technology, biotechnology, agriculture engineering. Earlier we were having a home science in order to articulate the knowledge in the mindset of a farm woman. Now we have converted home science into community science. We have got a food, nutrients and dietetics. We have got a veterinary science. So likewise, we have got 11 discipline. Now 3 more discipline has been added. One is a AIT, means Agriculture Information Technology. Where we have tried to articulate all component of a digital technology, starting from artificial intelligence, even a component of machine learning and deep learning we have imparted. How to use of Internet of Things, especially when to irrigate, how to irrigate, how much to irrigate. Every information one can easily collect with the help of a IoT based on things. Even we have tried to given a course related to big data analytics, means how to check the traceability, even means how to make a digital payment, and how to go extensively for content segregation, which is essentially required in our agriculture field. Similarly, we have introduced the course in AIT. We have introduced the course of a big data analytics, because in our agriculture we have got a lot of data. Only thing is that the accuracy and you can say precision and reliability of data is a big question, because from different source we are getting a data systematically and scientifically. Measures are not being adopted in the time of collecting of data. That's why we people are not in position to reflect a right position. Otherwise, we have got even augmented reality, virtual reality, mixed reality, extended reality. We have introduced these all component in our agriculture in a AIT. Even wireless sensoring, means things how you can measure the moisture content periodically, and you can easily understand that at what time your crop will start um, wilting, and then you will see that this is the right time. Even maturity indices you can calculate with the help of wireless sensor. So we have introduced in AIT a new course syllabus is introduced. Similarly, agri-business management at undergraduate level, because in a market, intelligence is one of the important component. If you see, in a government of India has introduced all 3 law, but unfortunately this has been withdrawn. I don't want to go its genesis and its why it has been withdrawn, but I would like to mention these are the really new the new light to our agriculture scenario. But unfortunately, maybe there's some other reason. But basically, where we in under this law, essentially a provision was kept to identify a new market, and in a market intelligent terminology which is called as a OPD is very popular. OPD means O means a opportunity, of course, new. P means how to penetrate inside, and D means how to develop it. And this is required in agriculture. We will have to identify a new market. As far as the profitability is concerned, as far as the remuneration is concerned, we are supposed to identify a new market. It is not that we will dependent on the old market. New market, then penetrate in this case, well, develop this. That was a reflection of government of India as well. They were interested to, even they have introduced a new mechanism of ENAM, Electronic National Agriculture Market. Means anytime, anyone and anywhere you can do the transaction. Even you can purchase or you can sell the product. That was a really a unique thing, and everything was so transparent under this ENAM. So basically in agriculture education, these 3 courses, AIT, ABM and REE, that is Renewable Energy Engineering. That is also one of the course, because in agriculture engineering, when we are talking about energy availability, and when we are saying that next you can say that only 10 years your LPG gas will be available, and we will be having a lot of problem and in future. That's why there is a need to identify some alternate sources of energy or renewable sources of energy like solar energy. And you must be knowing that government of India has decided by 2022 itself, within this year, more than 1,75,000 megawatt of electricity we will produce with the help of new and renewable energy sources, including renewable energy. So that's why a new course of renewable energy engineering at the BTech level we have started. And this is a very good course, because we want to give a impression that how students may have a idea of a energy availability at their farm itself. Because centralized electricity generation, it is not a right option, and there may be a lot of chances of a theft, and all problems are there when electricity is starting from a generation point until it reaches hardly 10% we are harnessing. And that is the reason why most of the electrical supply agencies, they are under deficit. And that's why there is a need to generate the electricity, that is a decentralized electricity generation with the help of renewable energy. Means at the use point itself, you kindly generate the electricity. You may have a solar photovoltaic system, solar thermal. Even the right example of your solar energy utilization is a greenhouse, whether it is a forced convection or natural convection, or it is a net house or glass house or polythene and walking through. Everywhere you find it is a right explanation of a renewable energy, or that is a solar energy. You can increase the photosynthesis process with the help of a solar energy. So that is a basically things. And here I would like to mention that a Indian Council of Agriculture Research is responsible for quality reform. Interesting thing is that in our country, agriculture is a subject matter of state. State is supposed to see the welfare of different colleges and university. That's why purposely I have mentioned state agriculture university, central agriculture university, central university with agriculture, and ICR deemed university. So we have got as I mentioned 64 state agriculture university, means where state component is available, because agriculture is a subject matter of state. But central, they are responsible for quality reform, quality assurance, and quality, you can say, refining, fine tuning of agriculture education syllabus, and making or rebonding the system, and reframing the whole structure. That whole thing is being meted out and through the central government. And that's why central government has given a very good a document, minimum requirement for starting of agriculture college, and systematically they have mentioned what type of quality reforms is required at the end of your agriculture colleges, what type of governance is required, institutional framework is required. Third thing means what type of a competency in the mindset of teachers is required, what type of a talented student is required, even what type of infrastructure is required. Each and everything is a very systematically mentioned is a minimum requirement. And here basically, as far as the syllabus is concerned, I mentioned fifth dean's recommendation is in position, and now sixth dean's recommend this committee is also formed. It is in position, and they will definitely conclude a new subject matter, and according to new education policy of 2022. Here basically, and now agriculture education will turn around the forward ABCD. ABCD. A means adaptive learning, more personalized, because the syllabus of agriculture and horticulture, it will differ from the forestry and fishery. Every subject has got its own importance. So adaptive learning, more personalized as per requirement, we will have to focus. If in some courses, say example in agriculture, we need 25 different department, but in fishery we need only 30 department. When we go to the agriculture engineering, we need only 5 department. When you go to dairy and food technology, we need only 5 departments. So if you compare with agriculture, you will feel. So that's why adaptive learning, more personalized. That is A. B means a blended learning. Blended learning means a more practical approach. Because without practical exercise, if you go through, and I'm sure that today I have not presented in any things in the form of a slide. So maybe possible some student will not go in position to grab and to understand each and everything. But those who are already in the picture as far as the education is concerned, they will understand. So basically, when we are saying blended learning, this is not a online and offline learning, but it is a theoretical and blending with the experiential learning and your practical exercise. That is a blended learning. C means a cooperative learning. Cooperative learning. Interesting thing is that in agriculture education, not only SWAM, not only ProCool, not only MOOC, that is massive online open courses. ProCool means a promoting cooperative learning, and basic idea of this all cooperative platform is that anytime, anywhere, anyone can understand the things, and very systematically. I give an example. If you are interested to make a value added product from milk, if you go for a paneer, or if you go for some sundaes and other manufacturing this, then you will have to undergo at least a 5 to 6 hour your practical. But when you will see through the online your video, then within a 15 minutes you will understand all unit operation. So that type of a component means anybody, anywhere, even if they are sitting in their class, in their bedroom, they can see that how to prepare a paneer and koya out of milk, and out what are the different unit operation as far as the modern irrigation is concerned. So that type of arrangement we have made in a through a pro-cool arrangement. Pro-cool means a promoting cooperative learning. MOOC means a massive online open courses. Some is also one of the things. So basically, these all cooperative learning component will be added, and it is already available, but in more refined way it will be added in future. And fourth important component is digital intelligence. Means in agriculture education, essentially whether you are learning, you are a dairy technology student, or a fishery student, or a community science student, you are supposed to have some understanding of digital technology. You should not go deeply what is the algorithm is used, how to prepare an algorithm, especially in artificial intelligence, and what are the different component of machine learning and deep learning. But they should understand how to use this algorithm in order to see the crop monitoring, in order to see the what will be the health of a crop in a subsequently 1 and 2 days, and then just to understand that how you can prevent the attack of insect and pest, and then finally you can assess and you can forecast this will be the end. So for that artificial intelligence is required, and that's why D means a digital intelligence will be imparted. So in agriculture education, it is expected these all four components very systematically we will arrange. And ICR used to give a lot of support to the state agriculture university, not only for education planning and development, but for human resource development, for human resource management, quality assurance and reform. Everywhere it is there. And number of new courses has been added in agriculture, frontier area of agriculture, like your conservation agriculture, precision agriculture, high-tech cultivation, vertical farming, aeroponic, hydroponic, even how to make use of a protected cultivation. And you will not accept my statement that our student, if you see agriculture engineering student, they have made a wireless sensor arrangement from their mobile itself. They are getting information that what is a scenariotical representation of different your nutrients in their soil. We say periodically they can judge that what are the losses as far as the fixed carbon is concerned, what are the losses as far as the nitrogen is concerned, in case if they are going for flooding. And even during the process of irrigation, if they are going for a drip irrigation, how much they have already irrigated in terms of centimeter of water column. That type of sophistication our student has made with the help of wireless sensing. Even we have developed a robot which is available in our university, and which we used to demonstrate to the farmers. Farmers is getting a information that which particular crop I will have to grow, and how to maintain the things, and at what time I am supposed to irrigate, and what will be the right period to remove the to attempt for interculture operation. Even recently we have procured a drone, and you won't believe that generally for completing a spraying for a 1 acre land, we were spending more than of 3 hours, and this process was a quite cumbersome, because farmer is keeping the whole Napscase sprayer on their shoulder and going to field and doing this exercise. But now within a exposure of 3 minutes, with the help of this drone, we basically are doing the exercise. It can carry around 12.5 liter of our insecticide, and uniformly it can spray. And the point from where it will start, that type of a algorithm we have made, that after completing, when it will become empty, then again it will come back to the original position. So that type of sophistication. So because of education only, we have increased. And here I would like to mention, because it is already 2:48 and 2:50. I was told that 50 minutes I will have to speak and 10 minutes will be the for our discussion. So I would like to conclude by saying I am really proud of. I have seen agriculture education very closely. When I got a opportunity to work at national level as well, and at 4 different places, I worked as a vice chancellor, and that's why I can say that our agriculture students has got a very good functional skill. Functional skill means it is a professional skill they are having. Means what to do, how to simplify the system, how to increase the efficiency and reduce the losses. That type of tips they are aware. Second thing is a soft skill. That you can say specialized skill. Under these students, because they are not a technical student, but they are aware that how drone are to be used. They are aware that how wireless sensor are being used for the purpose of this one. And our basic idea is to give us some basics and some practices in their mindset so they can use it. Recently, when we have procured a drone, our student they were fully trained. Even our staff they could not get the information. Even they were hesitating how to operate the drone successfully. But our few students from the second year itself, they started flying their drone in a very systematic manner. Now what we are doing, whenever there is a call from any farmer from any village, we are deputing our students throughout the day. They are going and they are doing this exercise. So that type of a things we people are maintaining. So basically they have got a soft skill, functional skill, soft skill, and third thing, very interesting thing is a innovativeness and creativity. And fourth thing is a entrepreneurship, because as I mentioned that we basically what we have done, we have given a right component of a trading. We have imparted not only skill but how to articulate the skill with a scale and speed. That type of component we have introduced. And fifth very important quality which our students is having, that is a contact outside of their institute. Very systematic arrangement is made at the national level, and thereby our student has got opportunity. They can easily go to the Punjab Agriculture University or Tamil Nadu Agriculture University, and frequently these students are getting an opportunity to visit to their different places, and that's why they have got a very good arrangement as far as the contact outside of the institute is concerned. And with this five of quality, I can say our students are really they are hope for this country. And in case if they will start their own business, I'm sure our GDP contribution from 19.9% in present context, it will enhance to 25%. And because if you see that even our policy maker, they have suggested that in a 5 trillion our economy means a 5 lakh crore dollar, if we our economy will enhance, and they have suggested at least 1 lakh crore dollar we are supposed to generate from the agriculture. And I'm sure with this complete capacity and quality, our students will be in position. So we have got a very good future of agriculture education in our place. But only thing is that as per the recommendation of new education policy, we will have to increase our capacity and quality as well. And we will have to solve the different problem of a farmer as well. For that, we all are ready. With this word, thank you very much for giving me this opportunity. And I have completed 50 minutes. So now, if you want to ask any question, I will be happy to give answer. Thank you very much. Thanks a lot to all of you.
Yeah, thank you very much, Dr. Rathore. So it's really important because in last session also, some participants were asking about in fact, questioning about the education of the agriculture domain. And you have nicely pointed out all the points related to agriculture education, the policies the government of India is designing, and the practical aspects which you have covered for the agriculture domain, and how course curriculums are revised at the government level, at the research level, research organization level, the industry level. So with this, we conclude the session, and now a few questions. Participants, please, any questions? Dr. Karthikeyan, any question?
So it was a very interesting talk, and you have given a very comprehensive view about the education system. And it was like how they are getting the industrial training and also getting acquainted with the latest technologies. Regarding Central University, just I want to know, already 3 are there, but a few more are in planning. Is there, or country right now only 3 Central University will be there?
Thank you, Dr. Milind. First of all, I would like to appreciate your effort, because when Madam Tiwari came, she has mentioned about your interest in a multidisciplinary activity. She mentioned that you are contributing in one of the research projects. So I was really happy to see your activity, because we will have to expand our activity only through this means. We will have to enter in a other domain as well, but with their permission. And we are supposed to contribute our basics and our knowledge and skill as well. So thank you, and I would like to appreciate your effort. As far as your question is concerned, Government of India, basically what is happening, we have got at a central level, there is a constraint of money. You must be knowing, since you are working in ICR. So very limited amount goes to our agriculture education. Major proportion we are sending it to the research, because we are expecting something new in our applied research and basic research as well. And second important component is that the Government of India has already established 731 Krishi Vigyan Kendra. So lot of amount of money is going towards their side. So limited amount is going, and since agriculture education is as far as the reform and assurance is concerned, it is part under the hand of a central government, whereas as far as the recognition is concerned, as far as the starting of any course and any university is concerned, it is a part of a state government. That's why Government of India is not interested to expand the central agriculture university, but they are interested to strengthen the leadership at the state level. They are interested to have some development grant in terms of, you must be knowing that when NAHEP, National Higher Education Agriculture Higher Education projects were funded from the World Bank, the basic objective was just to train our students so that they can become, it was having a three important component. One was IDP, second was a caste, and third was a innovative grant. So all the basic objectives of this all program was that for undergraduate student IDP, for postgraduate student that was a caste CAAST, Center for Applied Agriculture through Science and Technology, and for under IG innovation grant. So basically Government of India is thinking to strengthen the leadership quality at the state agriculture university. They are not interested to increase. If I remember when I was a DDG education, at the time the proposal for state conversion of state agriculture university into central university was there, and since our agriculture minister was from that place only, and when we have seen this different activity of our this Samastipur Bihar agriculture state agriculture, previous it was state agriculture university. Later on, when we have seen an high quality team was deputed, everybody has recommended that we should reshape this university. So there is a some way it was discussion. Even when I was there, our chief minister from state from Rajasthan, she has floated the idea of that let us convert one of the state agriculture university in central university. Nowadays, a proposal for conversion of Pantnagar into central agriculture university is under consideration. But I would like to mention that it is not worth to have a central agriculture university at every places, but it is worth to convert and to strengthen our state agriculture university so that we will be in position to get fund from central government, even from federal government, from state government. So that's why state government has got a more opportunity, and our state government has got a provision of a land grant pattern. When we are talking about land grant pattern, we say 50% of a grant should be provided from the state or from the central, and 30% from the state, and 20% of the grant we will have to produce in the form of a revenue. So that is a land grant pattern. If you remember, in 1960, when first Pantnagar University was established, in 1962, Hisar Agriculture University and Punjab Agriculture University were established. So basic idea of this agriculture university was on land grant pattern. So I would like to suggest that we should not encourage the people to fight for a central agriculture university, but instead we should have a state agriculture university and should avail the benefit and facility from all possible corners. Thank you, Dr. Milan.
Thank you very much, sir. It was very interesting to hear you today, and we will be looking forward for a future collaboration in this field.
Thank you, sir. Thank you.
Good afternoon, sir.
Good afternoon.
Dr. Om here. Sir, you mentioned about the subject AI, agriculture with AI and machine learning.
Yeah.
Sir, what you cover in that subject?
Yeah, basically, as far as the agriculture engineering, agriculture engineering undergraduate course is there, and as per the agriculture as well, we have given a subject of a digital technology. It does not include one component, but all 10 component, means a artificial intelligence, some thought, some thinking on a machine learning and deep learning, and especially how to use this AI in a different, means how you can have robotics, autonomous and indigenous arrangement, and what are the different application, because in China more than 23% of their interculture operation are being meet out from the robot. That is a basically the things. And similarly, in a drip irrigation, sprinkler irrigation, and new irrigation technique which is there in position, that is a sheet irrigation. Internet of things is required, means when to irrigate, how to irrigate. That component we have introduced. Some component related to big data analytics we have introduced. Some component of blockchain technology. It has got a 4 component, different component. I still remember because I was there at the time when this course syllabus was formed. We have included blockchain technology just for checking the traceability, and even how to make a digital payment and how to go for content segregation. That component we have added. Wireless sensors component we have added. Even a big data analytics component we have added. So all 11, even the concept of a image processing, like your drone, and in a even your augmented reality, virtual reality, mixed reality, and extended reality component. So some courses means just to understand what are the basics and what are the application. Fundamentally, we have not suggested that just to write a algorithm for a particular purpose. That was not our motto. But our motto is that a student should understand what are the basics of digital technology, all technology, and how we can apply this one. That is a course we have done. And in a AIT, Agriculture Information Technology, we have given a major stress that how this all digital technology can be useful for increasing production and productivity and profitability in agriculture. And the 4-year course is designed in Anand Agriculture University. This course is running successfully, and some new university, some more university has also identified, and they are also interested to start. I was also interested, but later on our government has suggested not to go for exclusively for agriculture, and we have started AI and data science, DS, AI and DS. So we have started BTech in artificial intelligence and data science. But we have tried to given this how to use AI and DS component in agriculture. That was basically our emphasis. But that our final award degree nomenclature is a BTech in AI and DS. Thank you.
Thank you, Dr. Rathore. So next speaker is waiting. Thanks a lot, Dr.
Thank
Rathore,
you.
for
Thank you,
accepting
madam.
our invitation and giving so much time. Thank you very much.
Thank you very much.
Thank you. So we have a next speaker, Dr. Koya. I request Dr. Ayan to please kindly introduce the next keynote speaker.
Thank you, Dr. Neha. So, hope I am audible.
You are
Yeah, you are
audible.
audible.
So, it gives me immense pleasure to introduce Professor Koyama from Gifu University. He was my past mentor. I was a JSPS fellow in his lab. And Professor Koyama is a senior professor and advisor to the Vice President, International Planning in Applied Biological Sciences at Gifu University, one of the old national universities of Japan. Dr. Koyama did his PhD from Tohoku University, Sendai, which ranks 76th in the world. Professor Koyama has been working in Gifu University from 1991 at different positions. He has also worked in foreign universities and institutes like University of Alberta, IIT Guwahati, and University of Delaware. He won the Outstanding Young Scientist and the Outstanding Scientist Awards. He is member of the Science Council of Japan and is the editor-in-chief of the journal Soil Science and Plant Nutrition. Thank you.
Thank you
Professor
very
Koyama.
much. Yeah, thank you. Nice to see you again. Yeah. So may I share the slide? Just a minute. Maybe. よいしょ。I'm very sorry. どれだこれか。んこれか。Can you see this? Data-driven plant science and green biotechnology. Okay, thank you. Thank you very much. Thank you introducing me, Dr. Satkan. And I always call him Ian. He's typical talented young scientist of India, for me, at least, anyway. And as he introduced me, I frequently visit India because maybe some people knows Gifu University and IIT Guwahati started the joint degree program in the food science and technology. We will jointly educate the student of Japan and India as well. And today, so I will not talk much about the science, but I want to introduce the recent trend of Japanese research concept, in particular focus on the green biotechnology and also the bioeconomy. Maybe you know the well about bioeconomy because in this sense, I think that India is the most advanced country, shifting from the fossil fuel-based economy to the bio-based economy. And I will little introduce our experience in the data-driven plant science. But first, I will introduce green biotechnology and bioeconomy. So I think green biotechnology is a common word for most of the student studying in the plant science. It's just a biotech crops. But actually, now it become more important than before because many countries started growing biotech crops, including India and Bangladesh. And also Japan, we recently started growing the biotech crops. And also that is good reactors for synthesizing the pharmaceutical substances or paper or biofuel as well. So it's quite important. It again becoming quite important. And today, so I try to introduce three topics. Divided. My talk is divided into the three topics. The first, Paris Agreement and bioeconomy. It's an upcoming new concept for at least for Japanese, and but it's very popular concept for the European peoples. And it's interesting because the bioeconomy, even the former president of the United States, Mr. Trump, he rejected the agreement of the Paris Agreement, but he accepted bioeconomy because this is quite important for the in the sense of the business as well. And later, I will introduce the green biotechnology and data-driven plant science. sorry, freeze. Okay, so can you hear me? I am.
Yes, yes.
Oh, okay. I'm sorry. Uh, Maybe you know the circular economy. The left side is a linear economy. It's an old style. Mass production and finally making the big pollution and just making the common stuff very large scale and then dump it. And it's a old style of the industry and the economy. And actually, the United Nations try to introduce a circular economy based on the recycle and reuse. And so every product should be designed for good for the recycle and reuse. That this movement was started in late 90s. And actually, just a few years ago, before the Kyoto Protocol, that was the first agreement, global agreement concerning the carbon emission, carbon dioxide emission. That was 2005. And now, so the new concept, the bioeconomy, become very popular political scenario for European country and some of Asian country, including Japan. But as I told you, so it's very interesting because the US also tried to introduce bioeconomy. It's because maybe you know this is the one agreement, uh, global agreement for struggling with fighting with climate change. And so United Nations, I captured this information from the homepage of the United Nations. And maybe you know the climate change can affect our health, ability to grow food, housing, safety, etc. And here it's very interesting. So this action, climate action, includes health issue, so well-being and also agriculture, grow food. This is the main focus of the United Nations action of the climate to struggle with to fight with the climate change. And here, left side, so just read this red font, red highlighted sentence. While a growing coalition of countries is committing to net zero emission by 2050, about half of emission cuts must be in place by 2003 to keep warming below 1.5 degree. Maybe you know we have to the United Nations and the Paris Agreement recommended us to keep the global warming below 1.5 degree. I will introduce a couple of papers that support this scenario. Okay. Oh, okay. Here and the bioeconomy. It is mentioned in the Food and Agriculture Organization of United Nations. So FAO, it's have the responsibility for agriculture and food production globally. The bioeconomy can be defined as the knowledge-based production and utilization of biological resources, biological processes, and principles to sustainably provide goods and services across all economic sectors. It's quite interesting because FAO stated that this bioeconomy based on the agriculture that should contribute to other economic sectors. So utilization of renewable biomass and efficient bio processes to achieve a sustainable production. And also the utilization of enabling and converging technologies, including biotechnology, integration across applications such as agriculture, health, and industry. It's quite, I think it's quite a new concept because FAO was already focused on the agriculture, but now, so they are start focusing the health issue and also the contribution to the other industry. Okay. And here, I just want to show this that the one of the very famous data for the peoples claimed the global warming. So here, this is changes of the temperature of the world, and actually last 1,000 years, you can see here it's 1,000 years level, so it means 10,000 years. Only just 10,000 years, the global climate became the stable, and so that's why the we started the agriculture-based civilization. And but now the peoples, human is the most serious factors for affecting the this type of very stable condition of the earth. And it was defined as the Anthropocene. This is one of the geological epoch. And actually, I didn't plot here, but global temperature jumped last 50 years, 2 degrees of the global temperature increased. So, any type of this type of argument focused to reduce, to keep the global condition that doesn't heat it over 1.5 degree. Okay. And this paper was published in the PNAS 2018. It was he's the first author of this paper, Dr. Stephen, from the Global Resilience Center in Stockholm. And by his group scenario, 1.5 degree increase of the temperature, it's melt the ice sheet in Greenland, and it's increase the absorbance of the heat from the sunlight, and it's induce, for example, the next step, for example, the Indian summer monsoon. Your monsoon will be affected by the global warming. And finally, if we cannot manage the global warming situation, it finally melts the ice sheet in the Arctic region. So, and also this paper reported different, very simple but very interesting data. So those groups propose the concept of the planetary boundaries. This is one scenario of the world situation, describing the world situation. It's divided into the portion like biodiversity, and here you can see the phosphate and nitrogen circulation in the world. And in the green color indicates each the boundary, so limit of the less impact to the global condition. If that become the yellow or orange, it gives a big impact for the sustainability of the Earth. So I'm sorry, I'm not the environment scientist, but this paper is now very, very popular for Japanese scientists as well. And for example, you can see the biodiversity and the circulation of the fertilizers, phosphate and nitrogen. It is might be the very serious condition for the sustainable if we think about the sustainability of our system. And from this paper, this is one of the scientific paper supported the Paris Agreement, but I think it provides a bioeconomy concept. It's better than the circular economy. And here you can see the last year, 2021, European Unions have one conceptual, provided the conceptual scenario of the bioeconomy, future transition for the bioeconomy towards sustainable development and climate neutral economy. So here, it's quite, I think it's quite interesting explanation how society and government work together for keep the sustainability of the planet. And it's they decided if European countries follow this scenario, it's a collaboration between the people and the government making the policy, both making the policy by the government and action by the peoples. This can only work to protect the planet condition. Actually, Japan also recently introduced this concept. Last year, we decided our government introduced various new policies for carbon neutrality and climate neutral concept. But also, it includes education. So higher education and middle education from the high school to the university, we start teaching this kind of stuffs to the student, like this. And actually, please remember that, but I'm a scientist. And for me, it's quite very good chance for the agricultural research. Because bioeconomy is, as I introduced you, it's based on the agriculture. And the last year, there was another very important meeting in the United Nations. For example, they analyzed the current situation of the food consumption in the global condition. And actually, we have already produced sufficient crops if that was delivered to the all peoples. But maybe you know the food loss and also the animal feed that gives very, very serious impact, negative impact to the food consumption. And also, it's that the animal production process is a very big sector for releasing the carbon dioxide. And also, 20% of the starch I'm sorry, crops is produced, converted to the sugar and starch. And this induce non-communicable disease. So this is metabolic syndrome. So actually, now our agriculture system, including the food sector, if we consider the food sector, our agriculture born the problem of the carbon emission, and also it is not optimized for maintaining the good health for the human. And last year, I'm sorry, and you know that the carbon footprint. For example, the producing this is the index how much carbon dioxide was emitted, produced when we make these food, 100 grams of these foods. For example, when we grow beans and nuts, it's just 1 kg or something. But when we convert it to the beef, in the average, 100 grams of the beef production release 7 kg of carbon dioxide. So we can convert all food to the carbon dioxide emission. And for example, now Japanese government as well, but the European country introduced the carbon footprint should be given in the package of the food product in the market. And for example, the consumers will choose the eco-friendly tomato, or for example, even the same tomato, if that produce more carbon dioxide, maybe some consumer will avoid purchasing that product. So I think this concept is also, if your Indian system can introduce this type of calculation for making the food product, it will give the big benefit, potential benefit to the farmers, adding the value instead of the organic farming. And here, anyway, so this type of meeting was held in last September in the United Nations, and they finally designed the planetary health diet. I don't tell much about this, but this food is good for the sustainability for our system, and good for human health. Anyway, I think as I told you, so I am plant scientist and worked last 40 years in the agriculture sector, and it's a good concept for making the agriculture more high affinity to the other industry, but also it is a good chance to increase the income of the farmers if we can support them to introduce this concept to their farming system. And I'm sorry, the next example of green biotechnology, it's my expert area, so it's much easier to explain you. So it's a green biotechnology is used in the breeding of crop plants and also producing the bio-based production. And for example, conventional biotech plants that become very popular globally. And I'm sorry, this statistics, this graph shows time changes of the how much hectares growing that were used for growing the biotech crops. And in late 90s, the American and the Canadian start growing the biotech crops. But 10 years ago, it has already started growing in the developing countries. And actually, the more amount of the area of the developing countries are growing biotech crops than industrial country. So now the biotech crops, I think that feeds the many people in the globally. And here you can see, for example, this is a good example. And in Bangladesh, recently introduced BT brinjal. And but actually, it's a first commercialization of the vegetables for using the BT technology. BT toxin, BT toxin is produced by the bacteriurensis. It kills the caterpillar, and it's not toxic to the human. And but it's all used to only the grain crops. And but the Bangladesh started using the brinjal, and I think this technology will be more expanded to the different crops. And a similar simple approach have already tested in the various genes and the various aspects. And left side, Professor Safu in IITG, he established the insect-tolerant cowpea seed that by introducing the alpha amylase inhibitor protein from the common bean. During the breeding process of the cowpea, that this protein was removed, irregularly removed, and the cowpea become very sensitive to the bugs in the during the storage. And just introducing the common bee protein, it protects the seed during the storage. And our group introduced organic acid transporter in the root that release beneficial bacteria from the soil that can activate the plant immune system, that can reduce the pathogenic bacteria problem. And the right side, the left side is transgenic, introducing the bacterial phytase that can solubilize insoluble phosphate in the soil, and introduce transgenic tobacco can grow better than the wild type. So these are all very simple approach, but I think this technology can be utilized to other crops for for improving the growth and the yield of various crops. And it's a recently this concept is more utilized in the for the improving the stress resistance, and it's quite interesting because the maybe plant scientist and student may know this photosynthetic system. And I'm sorry, that today, so or maybe the information peoples and computer peoples join this symposium, but I'm sorry, I will try to introduce this. And actually, the right reaction, it has a high risk producing the loss that disturb DNA and protein. And so plant shall balance, keep the balance between the light reaction and the dark reaction. Dark reaction produce a carbon, starch, fix the carbon. Okay, and in the usually in the higher plant have the photo respiration, that is a rescue system to reduce the risk of the loss production during the highlight stress. But here, you can see here the this process consume CO2, so it means the even photosynthesis produce a starch, but the photorespiration that make the loss of the carbon. And recently, one group applied very simple idea, introducing the bacterial genes that can skip this CO2 carbon loss by introducing one gene to the chloroplast. And here, for example, Norke, this group introduced one bacterial genes into the potato chloroplast. And they found that introduction of the one gene improves the yield of potato like this. The left two lines shows the transgenic potato carrying the bacterial enzyme that can bypass the photorespiration. That is a protection system of the photo damage in the chloroplast. Left side, this is the conventional, the wild type. So it's a very, very surprising result. But recently, other groups also introduced a different protein to the chloroplast that can protect the plant photosynthetic system from the highlight stress. Again, here in the highlight, excessive light that produce a loss like this. That this is a higher plant system. You cannot see any rescue system in this machinery. But the some of lower plant and algae here, Chlamydomonas, it carry the rescue system that can reduce the loss production by releasing the excess energy using the flavotidine proteins. It's a algae system but not in the higher plant. And one papers here you can see flavonoid protein substitution for cyclic electron flow without competition CO2 assimilation in rice. So one of Japanese group introduced these algae proteins to rice, and they found that the introduction of this protein can reduce the photo damage and but increase the CO2 assimilation. So it protects rice production during the highlight stress. So I think one approach, the breeding of crop plants, make the resistant to the environment stress, and also improving the yield. I think we can apply more about that with the transgenic approach. And next, I will introduce a recent, very good example of the bio-based production by the using the crop as a reactor of the reaction. And usually the chemical synthesis become the very expensive when making the complex structure chemicals. But biological production that is not directly associated with, correlated with the complexity of the compound. And using this situation, I think that we can use the crops and also bacteria as a reactor for the chemical reaction. And for example, it's very famous example how difficult to synthesize the complex chemicals by the pure chemistry. In the 1970, collaboration between the Harvard and universities, and at least 91 postdoc and 12 PhD received PhD, and this is 90 different nations contributed international project, spending 12 years for totally synthesizing the vitamin B12. It's consisted of 70 steps, and the yield is less than 0.01%. So it's no value for the commercial production of vitamin B12. Even it is contribute to receiving the PhD for 12 peoples. And the commercial production is now used bacterial system for making the vitamin B12. Beside, this type of approach was recently achieved in the rice, producing the astaxanthin, this compound. Here, rice usually cannot produce astaxanthin. That is good antibiotic. Antioxidant that reduce blood pressure or the cholesterol or anyway. So it's very good for health. And the Chinese group, they introduce 4 genes. First gene is the GMase, and the second, that is bacterial gene, and two genes producing the color, and these steps, catalyzing these steps, were isolated from the green algae. And they introduced these genes together, and finally they succeeded to produce the astaxanthin in the rice grain, like this. And so I think the in the current technology can allow this type of approach for producing the complex chemicals in the crop grains. Okay, so I think that conventional transgenic breeding, for example, introducing BT, that have already a very established system, and so I think it should be more utilized in the various crops. And also the comparative genomics and synthetic biology-based breeding, it can introduce a new system to crops, and it's also give the opportunity to the agriculture become more good to for high affinity for the industry. And so finally, so I just introduced a few examples of the data-driven plant science. And it's quite interesting. Maybe you know the GWAS, or anyway, genetic analysis using the big data set. And North Carolina State University, Cornell, and the University of Applied Science in Norway, these peoples work together for introducing new concept for selecting the stress-resistant variety in Africa using the more real data, using the by the collaboration with local farmers. It's quite interesting. Data-driven, decentralized breeding increase prediction accuracy in a challenging crop production environment. And here, maybe I can skip this explanation of this figure, just taking the image in the farmer's field located in the various location in the Ethiopia. And actually these farmers selected 4 varieties from the pool of the genotypes, and grow these varieties in their field, and taking the picture and send the data to the center, data center. And the statistical analysis and machine learning, etc., it gives a model for the prediction. And finally, they found that actually the old approach, this Ethiopian project was carried out growing the 41 variety plus hundreds more were extensively evaluated in 4 common garden experiment. That was old style for the prediction. But sometimes it doesn't fit the field growth of the farmers' place. And this project was then decided to collaborate with the farmers in over 1,000 locations. But they request to the farmers selecting the 3 only the 3 genotypes, and growing the total of 41 wheat varieties, and taking the data obtained the different location and different condition together, and then calculate than them established genomic prediction data model, and they finally found that this type of approach is very powerful to set up the high accuracy prediction model for adapting to some region, like in this case, so for Ethiopia. So I think this type of the local approach is quite important, but also we need collaborate with the farmers. And I can skip this. And lastly, I just introduced a few slides of our research. Actually, so Dr. Sadkan and the collaboration with IITG, I studied, I characterized one transcription factor. That is very interesting transcription factor regulating the different trait by the single gene. And here, for example, the knockout of this gene transcription factor make the Arabidopsis sensitive to low oxygen. It is similar with some merge stress, but the knockout give the Arabidopsis more tolerant to drought. So it is it regulates antagonistically regulates submerged tolerance and drought tolerance. And actually, this gene regulates other trait as well. And here, for example, iron, he reported salt resistance and drought resistance regulated by sensitive to proton isotoxicity 1. That is the name of the transcription factor. But as a student, he introduced, he found the stop also regulates the low oxygen tolerance. This is a submerged tolerance. And from that, actually, the stop-1 also regulate the aluminum resistance and proton resistance. We were interested in mechanisms of aluminum signaling. So that was studied by the, for example, multi-omics study and comparative genomics and promoter bioinformatics, and also pyramiding and phenotyping, comparison of the phenotyping, and used EGWAS or as a approach like the chemical genetics, understanding this complex system. And finally, we recently reported one review paper in the Trend in Plant Science. And here, this is the title of the review, synergistic and pleiotropy of root phenotype of regulated by the stop-1. And here you can see, because this gene regulates various downstream gene, it's quite important for the sulfate uptake and nitrogen uptake and potassium uptake. So that's why these play a role in the various role in the stress resistance. So that's why this is regulated very important stress resistant mechanisms. But it's questioned as because the plant must optimize the response to optimize to adapt to the particular stress factors like aluminum. And here, for example, the combination of EGWAS and the chemical genetics, these can model the less aluminum signal transduction pathway and the aluminum signal transduction system. So it's all from our research, mostly all from our research, and Professor Dr. Sadkan, he contributed well to draw this picture. I'm sorry, that is just introduction, introduce our research. But I think the integration of the various genomics approach, genome science, genome-wide approach, it's quite important to understand the true nature of the plant response to stress factors. So, its conclusion, bioeconomy oriented research, I think it's very important for the plant research right now. And also the comparative genomics and this, synthetic biology, it's a very promising to introduce very good materials for the other industry from the agriculture sector. And I think the field data provide a new idea for breeding. I think that that collaboration work in Ethiopia, that is very, very impressive. And I think that type of local approach should be improved agriculture globally. So that's all. I'm sorry I took much.
Thank you, Professor Koyama, for your enlightening lecture and giving very interesting examples of different research worldwide,
Yeah,
and also introducing bioeconomy, which is the
yeah,
main global agenda,
yeah,
and how India can contribute to this very important aspect.
yeah,
So,
yeah,
thank you.
yeah.
It was a very interesting lecture, and I'm working on Soybean Research Institute. So we are also working on drought tolerance
Yeah,
and doing GWAS studies to identify SNP markers for drought and disease resistance.
yes.
So it is curious to know that you have worked on this drought tolerance in Arabidopsis and identified new genes.
Yeah.
So I was wondering whether that can be applicable to other crops, and it will be interesting to see in legumes also.
Yeah, I think so. I think so because, for example, the stopan system that is conserved among the all land plant species, including the soybean, and we also found the same aluminum response in the soybean with the Arabidopsis. So I think our basic finding in the Arabidopsis can be utilized for the soybean. But also, some of the lower plant species like algae and anyway, moss, these plant sometimes use different strategies. Just higher plant, including the soybean, don't employ that strategy. But I think that if we can identify that unique strategy in lower plant, I think that can be utilized for the improving the crop tolerance of drought more than from the screening of the relative plant species or the anyway from the screening of the mutant. I think that approach, the integration with the genome information of the most and lower plant, it's quite interesting. And I'm trying that right now, that approach right now. Yeah.
Okay. So thank you very much, and we'll be definitely be in touch with you for future research in this area.
Yeah.
Dr. Karthikeyan,
It was a fantastic presentation, Professor. I am coming from chemistry background, so
Yeah.
for me it is eye opener. So now I have to see the role of chemistry in developing new crops with high quality, sustainability, etc., etc. So I like your circular economy that there should be zero waste, whether
Yeah.
it is coming from farming or from industry or plastics, etc. So zero waste from linear economy to circular economy. I love that. So what is your recommendation or suggestion in one or two lines? What India should do next 8 years to at least start with circular economy in agriculture?
Yes. I think that India is the most, as I told you, is the most advanced country in the sense of the bioeconomy, because you have developed a very good system based on the biomass conversion to the various stuffs. And I think that for example, degradable plastics made from the biomass. That is a quite good scenario for your country, because for example, country like Japan, we don't have sufficient biomass, but you have the huge potential producing the plant biomass. So I think the biorefinery is a very good good yeah, so you have the very big advantage than the countries.
So you recommend that we should use the biomass for developing some sort of biodegradable polymers that can actually help to avoid using a normal plastic chemical plastics. So yeah, bio-induced plastics, instead of waiting 500 years to decompose, this will decompose as soon as we consume. So as soon as we throw, it get consumed in the it goes to the cycle. So this is what you are recommending. Yes, it's very important. So in second question related to that, so how to go about it using technology, AI, high performance computing, genomics, GWAS, eGWAS. So what is your one word recommendation or suggestion for the young people watching our program to take this research forward towards that?
I think the for example, anyway, born the money from the agriculture, the making the income of the farmers, that is very important for the computational agriculture, optimizing the income, increasing the income. That is very important. But in that case, so please adjust the production system for bioeconomy concept of the Europe. I think if your country seek to export that the high price materials, in that case, the minimizing the carbon footprint, that is quite important. So I think that young people should learn more about the economic system, how the prices are made by the market. Yeah.
Excellent. So thank you, Professor. Thank you, Milind, for giving opportunity to ask these very important critical questions to Professor. Ma'am, I think we should take this one as one of the recommendation from in the minutes of the meeting, so that it goes to the government, it goes to the universities, so that people take direction towards circular economy. Thank you, Professor.
Yeah, thank you very much.
I enjoyed.
Thank you very much. Thank you for inviting me. And good inputs. Thank you.
Thank you. Thank you.
Thank you.
Yes, Dr. Rajesh, can you hear us?
Yeah, I can hear you. Can you hear me?
Yes, yes. So we are very welcome, Dr. Rajesh. We are very grateful to you for joining in this program. And I would like to give just brief introduction. Dr. Rajesh Periniagam is currently a founder of a Cariosoft company based in US. He has done his BSc from Madurai Kamraj University, MSc from Hyderabad University, and PhD from National Chemical Laboratory, Pune. So we have been collaborating with him since a long time, and last 25 years he has good experience, international experience in academics and also in private company. He has worked at University of Missouri, Columbia, Washington State University as a research scientist, and also in a big agriculture companies like Dow Agri-Chemicals and Roche Biodiagnostics. So he has a vast experience in industry and academic both. So welcome to this program, and we would like to hear from you.
Thank you, Dr. Milind, for the nice introduction. It's a really nice meeting all of you. I think everybody is new to me except for Dr. Milind and Dr. Karthikeyan. I don't know, he still remembers me from NCL time. I can
clearly recognize the building behind you.
Yeah, that's why, I thought that you will mention that. Thank you.
I remember the time I spent over there. I love
Yeah,
that.
I loved this building so that I could not leave even after my PhD. Okay, yeah,
Of course.
thank you.
Anyway, let me share my screen. Please let me know if you can see my slides or not. Can you see my slide?
Yes.
Sounds
Yes,
good.
Rajesh.
Okay. Yeah, first of all, thanks to the organizing committee for inviting me to give this keynote address. I mean, once again, nice meeting all of you. And it's truly a privilege and honor for me to spend time with all of you today. When Dr. Nega reached out to me for the title or topic to talk, it didn't take much time for me, mainly because I have a great passion for agri-genomics because my career started using agri-genomics technologies for crop improvement, and until today I have been involved in agri-genomics. So that's why I wanted to share my perspective of my experience in this topic with all of you. And here is the outline of my story I'm going to share with you today. First, let me start with the introduction to give some global demands and scenario. Then I'll talk about the agri-genomics in the past. When I say past, what I mean by is it is a pre-next generation sequencing technologies. And then I'll switch gear to the current scenario, what's going on in the last, ever since the inception of next generation sequencing technologies, with just two specific examples. And then before I conclude, I'll touch upon agri-genomics in the future, where it is leading to. And agriculture, as most of you know already, play a key role, contributes a lot to the global economy. For example, it's estimated to be agriculture contributing 4% to the global GDP. And in the US, it's a 5%, and towards the employment, 10%, close to 10% of the employment is through agriculture and related industries. And the challenge is, I mean, we all know that the population is going to grow to 9.1 billion by 2050, which is currently from 7.4 billion. And among those 7.4 billion, one out of 7 people are undernourished. And the challenges are the crop growing lands are stagnant for quite some time. I would still say that it's not only stagnant, it's going down. For example, the place where I live right now, it used to be a cornfield. Now it became a residential property. And then after I moved in, I have seen several of the cornfield disappeared and became commercial properties here. And because of that, UN recommended or forecast that the food production should be increased by 50 to 70%. So consider the scenario like increasing the population and reduction in the growing land, crop growing land. So that means there is the only way we can meet the demand is by adapting to new technologies to increase the productivity in terms of the yield and everything for all the food crops. And while we are doing, we need to be cautious of the environment as well. So global ag sectors from academia, institutes, and industries, all of us are contributing to this global demand. And coming to specifically genomics, as you know, there are a lot of technologies that are contributing to the growth of agriculture. And in this talk, I'm just focusing on only the genomics. So the global market for genomics as a whole in life sciences is estimated to have to be at $22.7 billion, and it is going to grow in next 5 years with the compound annual growth rate of 19%. So within that, agri-genomics, if you take only that, it's currently estimated to be at 3.3 billion. And in the next 6 years, it's going to grow at the 9.7%. The major drivers for this growth is food demand, the one I mentioned in the previous slide, and also the rise of the population. So the goal of the whole agriculture is essentially to discover favorable traits in the crop of our interest, and we know that what is the governing factors for those traits. That is, the discovery part is very important. And I mean, we all know that variation is the one that causes that trait differences. But the bottlenecks many times are to find how fast we discover those genetic variations, and how fast we can integrate those traits to a elite variety to or any desirable varieties to improve the yield. So in this talk, I'm going to focus on those two topics only, but there are a lot more areas we can cover, but considering the time, I don't think I can cover all of them. So let's talk about how agrigenomics was in the past. So regardless of any technologies we use in the field to measure the phenotype for different traits, and various technologies we use for genotyping the population, the simple formula is the ultimate goal is to correlate both of them, phenotype and genotype, and to transfer either the agronomically important genes or improve the yield of the varieties and so on and so forth. It is a very simple formula, but behind each step, there are a lot of hard work involved in that. In the past, before pre-NGS, the lab workflow used to be very simple. Scientists prepare samples, extract DNA, they do the genotyping, and collecting the phenotype that comes as a parallel path. And then we combine both genotype and phenotype, do the linkage analysis, and then drive the innovations. Of course, in the genotyping, there are a lot of technologies involved, but that used to be a kind of parallel track. Like some of them, the community resource or something available, or scientists generate a small insert library and then screen the library to discover microsatellite motif or something like that, and then they design primers and then share with the community. So this used to be very simple at that time. Here, as you can see, these are the various molecular technologies that was used before NGS. And I was fortunate to use most of them in my career. But regardless, every technologies have different advantages and disadvantages. But to me personally, I felt that the impediment, that the bottleneck was we had only fewer molecular markers. And that means we could extract only limited amount of genomic information for downstream analysis. But on the bright side is we didn't have to have a computational needs much. And one scientist can handle all those markers. And we can combine, I mean, through collaborative work, we can use different types of markers to develop linkage map or QTL discovery and gene discovery and so on and so forth. So it was, on the bright side, it was simple and it's doable by one scientist like that. But currently present, with the inception of NGS, the dynamics has changed. So I'm pretty sure most of you must have seen this picture before. The reduction of, with the commercialization of next generation sequencing technologies, the cost for sequencing a genome dramatically reduced. If the market has followed the typical Moore's law, we would be sequencing one genome at the cost of $100,000. But with the commercialization, with the demand and all of them, from the 2007 time onwards, price reduced, the cost per sequencing genome got reduced dramatically. Now it is at the range of close to $1,000 per genome. The good news on that part is now we have the opportunity to see a lot more genomic information, and we have the opportunity to understand the organism at the whole genome level, not at the specific locus level. So that is a bright side, but on the downside is there is a lot of data in there. So because of that, the genotyping part, which I was referring to, that got expanded. Because even when the inception of the next generation sequencing technology, it used to be a kind of parallel track, but now with the last 20 years, last 15 years, it became part of the regular workflow of the scientists. So now instead the genotyping starts with the genome sequencing. Once the sample is prepared and genome is sequenced, and now we have so many options, first generation sequencing technologies like Sanger, second generation and third generation sequencing technologies. So because of that, we are generating large amount of data. Then that means that data needs to be assembled and annotated. And then once the data is assembled, then it needs to be, we need to have a platform to keep them at one place. And then we take the data and then combine that with the phenotype and do the molecular genetics analysis and then drive the innovations. So that part, we because of that change, the demand of what we need to handle those that amount of genome also got grown. So because of the dynamic change, what we did was we did NSF, National Science Foundation, USA sponsored study to understand what's going on in the field, what's going on in the scientist's life because of the changes because of the introduction of NGS into the common workflow. So we did, we talked to several scientists like all of you. And what we learned was how it affected was scientists' life was we found out that 25 to 70% of scientists' time on searching for the data, data preparation, and organizing the data and so on and so forth. So why that happened? Because the reasons behind for that time being spent on just data preparation rather than driving innovations these were the various reasons. The common concerns, the challenges what scientists were facing was they don't have proper data storage for their data. And it varies depending on the organization, but think of that. Data storage was a challenge. And even if there is a storage, there is no easy way to access and share the data with the colleagues. And also they needed, some scientists wanted to combine their own data with other collaborators' data or public data, and then use the broader amount of data set for innovations, but that was not easy. And even if there are tools available, it's not user friendly for scientists who are not computationally savvy. On the other side, because of the large amount of data, computational power is needed. I'll tell you how much power we need in the later time. And computational expertise. Not only the power, and we needed to have the computational expertise to drive all those, manage and then assemble all the data. And bioinformatics expertise and their time. In a typical organizations, bioinformaticians are not many in number, and they are challenged with their time because if everybody's going after them for, hey, can you get this information for me? It takes a lot of time for us. So putting all of them together, what happens is the data loss, because when the scientists leave the organization, the data is lost. And because of that gap between the data and the scientists, it drives the slower innovations and then eventually slower productivity. So all those learnings were the genesis of Karyosoft. So just I wanted to spend a little bit time to share with you who we are and what we do before I showing the examples. And we are headquartered in Carmel, Indiana, USA, which as Dr. Millen introduced. So our mission is to develop simple to use in-house genomics software solutions to help the scientists to transform agri-genomics data into actionable insights. And think of it like your own companion for your own data sets, whatever you want. So just I would like to share with you some of the recognition we received in the last few years. And I mean, 3 months back, we were recognized as one of the 30 startups to watch by World Agri-Tech Innovation Summit in San Francisco. And you can see additionally other recognitions as well. So what, because we understood the challenges from interacting with a lot of scientists and also my own personal experience, we developed 3 platforms that focus on that genotyping area itself in the part of the workflow. So one of them is a variance platform, which is for the variant analysis, how fast we can discover the mutations in your population. And then Variant Mining Studio is like your in-house repository for all your variance data. And we have a third platform is Loci, which is like your own customized GenBank. So the current way the data is mined is through command line, and it's used in single computers and only experts can do it. And then we circumvent the issue, that challenge, and then make the data easily accessible to the scientist, and they can access anywhere, anytime with a few clicks to get the insights. So our core capabilities, what we are capable of doing is we are good at cloud computing, and our software can be installed on-premise, and our platform is organism agnostic and highly scalable database. We are good at developing, and especially industry clients, they prefer data security. So since we install on-premise or cloud, their their internal firewall or their data, the security measures will be automatically applied, and it can be accessed anywhere, anytime, and also we can customize this platform. So these are the proven applications of our platforms to date, and gene editing in CRISPR-Cas and gene discovery, SNP discovery, and so on and so forth. So coming back to one specific example, first example I wanted to share with you is the variant analysis. As I mentioned in the beginning, the need is to, all the traits are governed by variations at the genetic level. So faster we discover the variations, quicker we can transfer to the field. With that goal, we build this variant platform. So like I mentioned, why discovering genetic variants is important and why it is challenging. And as I mentioned earlier, that the variation is the one gives the organism to survive better compared to others in different environment. And this helps the scientists to understand and improve the organisms genetically. But the current process of doing the variant discovery is time consuming, laborious, and command line based. Only it can be run with the experts, and involving multiple databases and so on and so forth. Few years back, variant discovery was a big task. But now, especially in the last 2 to 3 years, because of the COVID, the variant discovery got expedited and made, because of the COVID situation, but the flow was faster because of the COVID situation. But that is in the healthcare side, but obviously we can do the same thing in agri-genomic side as well. So this platform is basically, like I mentioned, it's organism agnostic and browser agnostic and device agnostic. And we developed this platform in collaboration with AWS and NVIDIA. So the input file is this simple. You need only 3 files to run your platform, run this variant analysis, reference file and annotation file and your sample file, which is the raw data. And the output file of the variant tool is automatically used to upload into the variant mining studio. So here, for this presentation purpose, I ran as one analysis for the soybean accession from NCBI. So this is the platform where you simply you have the flexibility to run single or multi samples, and you can start the project by naming the project. I started with soybean. And you can upload the data in so many different ways. One is you can FTP your samples, or if you are in cloud, you can upload the data into S3 bucket in AWS, or you can directly take it from SRA database. But I took from SRA database directly. And then the bioproject number is SRR 19693188. And unless you change any parameters, all those 6 steps are automated. But initially, we recommend you to run all of them with the default value. Then you can play around the parameters. And once every step is completed, you will get the timestamp as well. So that way you can go back and see how long it took each steps and how long it took from end to end as well. And you can submit as many jobs as you want, but all of them will be automatically go into the queue manager, and then it'll automatically take one after another as per the time of your submission. And those are the input format. And then output file, there are many, but I want to highlight just a couple of important files. One is the data quality report is automated, it's incorporated into the workflow, this is very, very important, because if you run a data with bad quality, and it is going to lead you to wrong innovations, right? So that is why this quality report is incorporated, and you can see for all your sample data, the raw data, how high quality or low quality and all of them. So you can make a decision to discard those type of data. And in addition to in addition to what you are seeing, that there are 11 different quality parameters for all your sequence data, and you don't need to run them separately before you run the variant analysis. And download device, you can each step generates a lot of files, and each one you can download, starting with your reference genome in FASTA format, and also with index file. Because if you are using any genome browser or something, you need to have the indexing of your reference genome. So this way you can use that to visualize your data. And you can download a lot of other files from every step. And most importantly, your SNP and Indel variant report. And BAM files can be downloaded, and VCF files with annotation. You can download them, and also you can download the VCF file as a whole, or you can download by chromosomes. And here is your the genome-wide report for the soybean. And for this particular line, we found more than 1 million SNPs and more than 170,000 indels. In addition, you get additional information like the impact, the mutation impact and effects, and where the mutations are distributed across the genome and all of them. But overall, other than that, the most important thing is for the end-to-end analysis, it took only 47 minutes. And but the data size is basically around 7 gig for both paired and sequences. So it's a smaller file size. That is why it took 47 minutes. So in order to see if this is one sample also, right? So we ran parallelly collaborating with scientists from Germany. We ran 4 samples of their rice samples with the total of 72 gigabytes file of one sample. So we ran 4 samples, each one has the size of 72 gig. And we ran through 3 different platforms to see the speed. So when it's all run on the cloud, and when we use the hard disk and CPU hard disk, it took about 48 hours for 4 samples in the parallel computing aspect. Then when we try to adjust the hardware, that still reduced that, but still it was much it reduced the time from 48 hours to 28 hours, but still it is 28 hours per sample, or 4 samples parallelly. So in our case, it came down to 4 hours. So with that, we did the same thing with serial computing as well, but overall 7 to 12 times faster than other platforms, and then that corresponds to cost. Because if you are running only shorter time, the cost also gets reduced. And beyond that, the time. If you are running multiple samples, it could reduce the time significantly for when you run multiple samples. So that is the serial computing also, meaning if we are running one sample after another like that, still it is a significant time reduction of our samples. So the output file VCF, when you download this file, typically when you open on Excel, it looks like this. And if you are looking for any information, it has a lot of challenges. Besides, when I said that it's 1.1 million SNPs, and Excel has some limit for the rows, right? If the total number is more than 2 samples, or more than 1.4 million or something, you cannot open the whole Excel file or whole VCF in one file. So it is very difficult to extract information from the gene of interest or location of your interest. So that's why we built this variant mining studio, which is a central repository for all your SNPs. So where I simply uploaded that VCF file that I showed you into this one. It took 15 minutes for loading all 1.1 million plus 170,000 indels, all of them. And I just looked for a specific gene, which is RPG1B, which is NBS-LRR gene, I believe. So I searched by RPG1B, and the result showed up because at that region, we had 20 SNPs. This is only SNPs, and then indels you had 2. And you can get all the parameters in a very user-readable way, like the chromosome number, position, reference allele, alternate allele, quality, and filter, and so on and so forth. And then additional parameters also, you can filter them by different other parameters. It's what you select. And also you can look at the annotation information. So this is that particular locus annotation information, what the putative impact and all of them, and where it is located and all the information, gene feature ID and everything. And also you will get the information on the genotype file, meaning for this particular sample, the genotype is homozygous ultra-allele. And if you are running multiple samples, all of them will be one below another with the genotype file, genotype of each sample for that particular locus. So you can simply download the mutation information, SNPs and indel information for gene of interest instead of downloading all of them. And like I mentioned, these are the 20 SNPs we discovered for that particular gene, and indels are 2. And like I mentioned, all those information, you can see the chromosome number, position, and everything. And here, annotation information, what the impact, where it is the mutation, all these mutations are discovered. So downstream gene variant and the effect is modifier. So these type of information will help you to choose a specific informative variants for your downstream work. I mean, the goal of all this analysis is to find out variations that has the effect, right? And then if it is a downstream gene variant, that is going to helpful. It's not going to be helpful. But if it happens in the exon region or one of the promoter region, and or any of the with the effect of frame shift mutation and so on and so forth, that type of mutations would be extremely helpful for downstream analysis rather than using millions of SNPs, because all of them are not going to be helpful for us. But we can bring down subset of the variants with the serious effect, and you can use that for your downstream analysis. So we made this variant platform available on AWS Marketplace, and also what the next step for this platform is. We are always looking for reducing the speed and reduce the cost, because that is the only way it will help scientists and everybody. So that is why we always look for new algorithms for improving the speed, and also for looking at the computational side via angles like computing orchestration, and also serverless architecture in AWS, which they also starting recently. And those are the technologies we are looking into to reduce the cost and also increasing the number of samples to run. And also we are working towards integrating this data, everything into our LOSI platform, so that scientists can retrieve not only the SNP data, but also the full sequence data of the gene, and then they can use it for gene cloning or other downstream work. And also we are looking into other data types like RNA-seq and so on and so forth. So where these, the output file, what you get can be used for your work is, if your goal is to discover SNPs or indels or mutations at the gene of interest, which is what basically, that's a fundamental of molecular genetics, right? And direct genotyping of core germplasm for, and then use the genotype data for association mapping. And you can choose specific SNPs and design a custom chip for your from your parental lines, and then screen those chips for your population screening. And if you want to design any PCR-based primers for the SNPs or indels region, and then use that for amplicon sequencing, there also this data output would be used for your work. And there can be many more. These are the few thoughts came in my mind. So now what I explained is so far is how fast we can discover variants, mutations in agriculture. But now how it can be utilized for breeding purpose. So here I would like to share with you one study where the next generation sequencing technologies, GBS, that was used in trait introgression for high quality conversion. So this work was done in multidisciplinary team involving quantitative geneticists and bioinformaticians, computational scientists, molecular geneticists, and breeders. So the trait introgression, we all know textbook definition is very simple, is movement of a transfer of a gene of interest from donor to the elite variety through repeated backcrossing without altering the performance of the elite line. So that means we needed to avoid any linkage drag, any drags that comes from the donor lines. So ultimate final goal is to convert your elite line, which is with the recurrent parent percentage of 98%. And assume that just leaving certain percentage, or more than 98% would be ideal. So if you want to have 98% or 100% very similar to your elite line, that means we need to have the genome view. We cannot look at the genome partially and then we cannot claim that we converted into 98%. So initially what we did, we analyzed, we assessed which marker, whether it is application of NGS is how effective and all of them. So we compared three most recent technologies like microsatellite based screening and SNP and GBS. So there were several attributes we looked into that and compared all different traits with the different types of markers. And then of all of them, the last three which are highlighted are the one prompted us to use GBS, meaning number of loci being analyzed, microsatellite markers and SNPs. And the SNPs, what I'm referring is, we had the core SNPs and the Infinium chip, and that was screened with the parental lines, and then we had those SNPs for all the parental lines. That is what I was referring as SNP. And similarly, SSR also the same, microsatellite markers. So we had core SSR markers, and then parental lines were screened. In the parental lines I'm referring is those were used in the introgression program. So the number of loci we could analyze with SSR was low, and SNP is also low, and GBS we could do that with greater than 1,000. And development cost wise, also there is a low cost GBS, and cost per analysis is also low. So those are the attractive attributes that prompted us to use that GBS in the trait introgression technology program. So the typical workflow with the SNP was, from the field, samples are sent to the lab. A DNA is extracted, and that needs to be transferred into multiple plates. It was analyzed individually. And then DNA needs to be dispensed, and then cocktail needs to be dispensed, and then low throughput genotyping was performed. And that was combined with the field data and perform analysis and made the selection, and then send that information was sent to the field. Then breeders advanced those selections to the next generation. So this was the workflow with the SNP marker technology when we were using at that time. The challenges were the volume of the samples, because if you are running just one or two projects, that is handleable. But when you are running multiple conversions, the sample volume is very high, and then tight turnaround time, 9 to 12 days, because plants are growing in the field. So we have to run analysis and then send the selections to them, and then so that before flowering time, they can choose that and pollinate and then drive to the next generation. And then a lot of manual steps. As I mentioned, the plate replication, liquid dispensing, and genotyping, and all of them involve manual steps. Those are the constraints on the lab side, but scientifically, many of the crosses, because when you choose donor, you don't have much of choice. Donor is most of the time it's fixed. You handle with one or two donor lines with the trait of interest, with the proper variation. But elite, we had a lot. So when you look at the genetic relationship between the elite parent and your donor line, majority of them are genetically narrow. So what that means is that gets translated to the number of polymorphic markers we could find in the specific cross. If we don't have a proper coverage, that means we are not seeing all the genome. So that affects the quality of the selections, quality of the conversions. And also cost per sample. And since we are running all the markers individually, lack of multiplexing add additional cost to that. So, for example, if you are not using marker technology, it takes 6 or 7 generations for you to achieve that 98% recurrent parent percentage. But with markers, we can reduce the time with 2 or 3 generations shorter. We can shorten that, so that expedite the variety development. So finally, that time we were looking for a cost-effective, time-saving, and informative technology to introduce trait of interest into a elite inbred without losing elite inbred's performance. So this is the pathway to our implementation. Initially we did proof of concept with fewer samples, and then expanded based on the results. We transfer to pilot studies, and then pre-implementation, and full implementation. So here is the in the workflow wise how when we compared the SNP based workflow and then GBS based workflow, we compared that and we saw that there is a lot of reduction in the steps. So these are the steps which I showed you earlier. And with the NGS, almost from plate replicator to data scoring, the whole steps are eliminated. It's all one-stop shop. And then directly you can go to the data visualization and make the selection and go. But there were a lot of challenges to smoothly integrate, transfer. I mean, when we were doing the proof of concept, there were several challenges in terms of how to run and how to get the GBS faster and so on and so forth. But regardless, we were successful on that process. So with with GBS, we got the opportunity to see the genome as a whole, and time saving by cutting down several steps, and cost effective is because of the multiplexing capability. So there is a that way we can multiplex multiple samples. That reduces the cost, and also we didn't have to go through that traditional parent screening of all elite lines through the Infinium chip, which was very costly at that time. And of course, once we learn that, this can be transferred to other systems like other crops. In fact, the one I'm explaining is, although you consider that as a general outline, but it can vary from crop to crop and system to system, but general workflow can be used across multiple systems. So this work was done in a 3-step process. One is a 4-step process. First, we selected suitable projects for the POC. Because it is a POC, we just took only 2 population. And one was narrow cross, meaning the donor and the recurrent parent both are genetically similar. And then we chose 3 generations of the backcross population from that cross. And another one we chose the white cross, meaning that recurrent parent and donor both are white. And once we selected, those samples were run through GBS. That's a typical common workflow. And ultimately in that one, we mapped 2.4 million reads to the reference genome. And only the polymorphic GBS markers, SNPs only were used for the analysis. The 3rd step was in the population, we saw many missing steps, missing data. And so we ran those analysis data through imputation pipeline. So basically that filled in a nutshell, it filled the gap in the population. As you can see in the population, the general diagram. And there were many missing data, and that got filled based on the parental genotype data. And on the right-hand side, the table, what you see is the numbers. That was mind-blowing. And with the white cross, with the SNP, we had only 99 to 96 and so many, only fewer markers. But with the GBS, the number went several fold higher. And with the narrow cross, which is important, where we had the limitation to see the genome very small, which was in the less than 50 or close to around 50. But with the GBS, we had almost 10 times or 8 times more than the SNP markers, and then all were spread across all the genome. So we were able to see the genome much better than using Infinium SNPs. So once the analysis is done, we visualize the data. This is the chromosome table, we call them, and the top number 4, what you are seeing is the chromosome number, and then the 20 to 30, 30 to 40, that particular line is basically 10 centimorgan interval based on genetic map that was pulled in. And then the number what you see is 4, 1, and all of them, those were the number of GBS markers in that interval. And in the table, what you see is it's like a traffic signal. Green is more than 80%, which is ready to go. And if it is less than 20%, it's brown, and orange is somewhere in between. So we always aim for the selections which is more than 80% at the first generation. It's a BC2 itself, and then that shortens the time frame. The key takeaway point is the purple color area, what you are seeing is we didn't see this entire region with the SNP markers, with the Infinium chip when we selected. We didn't have any polymorphic markers in that region, but with GBS we were able to get it. And imagine that if this is not there, and accidentally if I select this particular progeny, because each line represents one progeny. If I accidentally select this particular line, I am taking a lot of linkage drag into the next generation, which will take one more generation to clean it up. So that is the advantage of using, that is the advantage we felt that using GBS in the trait introgression. We were able to see almost the entire genomic intervals in all chromosomes. So that helped us a lot. And in this, there is clearly one line is in this chromosome is fully converted. So we looked into the other chromosomes and then see them and selected that one, that particular progeny to advance to the next generation. So of course, I mean, after we selected downstream work-wise, what we did was we looked into the performance in the field in terms of the yield and all of them, and made sure that that yield is not affected. And also we correlated the SNPs versus GBS with respect to, I mean, for both narrow cross and then wide cross, how highly they can be correlated. And the key learnings from this work was we saw that the conversions are high quality. That's very clear on that with the whole genome view. And time saving, because of the complete turnaround time from the field to the field back, we could reduce the time as low as 7 days. And with the GBS, we could multiplex, because initially the multiplexing was at that time 48. And then as we progressed, it went up to 1536 samples multiplexing. That it went to that high level. So that shortens the cost, that reduced the cost very dramatically per sample. So that was the key learnings what we learned through this one. So far what I have shared with you is how variant discovery made easy, and then how fast the variants can be introgressed to develop elite variety through backcross breeding with the trait of interest. The next few slides I would like to talk about where agrigenomics is heading to. So like I mentioned in the beginning, the cost per sample, sequencing sample is reduced dramatically. But at the same time, I mentioned that the data growth is exploding right now, right? That's growing a lot. And the cost-wise, what I mentioned is up to $1,000 per genome. I mentioned at the beginning, but 2 months back, one company from San Francisco is claiming that they can sequence the genome for $100. So, I mean, how feasible it is down. We are yet to see. But regardless of that, if the cost of the sequencing is as dramatically keep on reducing from $100 what later $10, who knows? But regardless of what direction it is going, we have the opportunity to generate a lot of data. And then how much? That is the question. So in this graph, the black line is the recorded growth. And the blue line, what you see is the typical Moore's law, meaning the data volume is doubled for every 18 months. That is a typical Moore's law. And if that record is followed, which is very conservative, and we are expecting by 2026, the data size is going to go more than 1 exabytes. Illumina predicted that the volume will be doubled for every 12 months. If that happens, it is going to be much more than the Moore's law. But the recorded growth based on that projection, it is even going beyond zettabytes. So it's very simple, very straightforward that the genomics data production is going to explode. It's already started. It's going to go big. And in this paper, they compared 4 domains of big data currently. The work was compared against astronomy and Twitter, YouTube, and genomics. And they focused, looked into 4 different phases of this data: acquisition, storage, analysis, and distribution. And as you can see in the storage part, by 2025, astronomy, which we used to say that, right? Astronomical growth, we call that, right? So the data is going to be only 1 exabyte, but Twitter and YouTube, which is our common life, but coming to that genomics, it's expected to require 2 to 40 exabytes per year, which is huge. So that is one question is, are we going to replace astronomical with genomical to describe phenomenal growth? We never know, but we are in that era looking at those faster growth. So like I mentioned earlier, in this talk, I'm focusing only on the genotyping portion of our workflow in the molecular breeding work. But genomics have different areas where we can utilize a lot more information and a lot more applications. And but the data volume, what I said, it's growing, is going to be overwhelming for us. But at the same time, if we manage it, if we do it properly, we get we can extract best information out of all those data. So in this talk, I mentioned only about the genomics data growth. It's going to, it is still growing, with the reduction in the cost, it is going to grow more and more. That is going to go. But in addition, I never mentioned anything about other omics data that can be utilized in the same workflow, like RNA-seq data, functional genomics, and metabolomics, and proteomics. And if we include microbes into our crop improvement, then metagenomics data, metatranscriptomics data, and so on and so forth. There is a lot more data is coming, being utilized already, but I didn't mention in this talk. And because of that, data is one area. In the future, it is going to grow more. Because of the reduction in the cost of the genomics data, the next is RNA-seq. Because at this point, RNA-seq cost is still much higher. Not everybody can afford. And but automatically that will also drive the force and then reduce the cost. So like that, every technology, every omics data generation technologies will reduce the cost down the road. Because of that data volume, per institution, infrastructure needs to be built in accordingly. Imagine our researchers' life without NCBI or GenBank, because we have that resource outside. So that is why we don't feel the pain much. But when it comes to when you want to have your own data from within your organization, the infrastructure needs to be built internally for storage, not only for the raw data, but also for the assembled data for easy access and get the information fast. And the computation speed, it's not just the data storage, and also we need to have the computation speed to extract the insights faster. And we need to connect the data to the end users, the users, biologists or scientists. For that way, scientists can get the insights and drive the innovations inside the organization, in their project or the organization. And then last, not only, last but not least is the analysis tool. There are several tools that are coming in that area of the genotyping part I was mentioning, the raw data to fully assembled data, that three-step process. There are a lot of additional new technologies are coming. For example, AI deep variant. And this deep, but it has the limitation is this can be applied only for the human data set, but not for other system. But if we use them, then we have the that tool also can be utilized in our regular pipeline for variant calling. And conversational language, to get the insight through conversational language. But now we are using iPhone and smartphone to talk to get the information. Why not we talk and retrieve the genomic information? Just talk to that our computer or data, say that, hey, how many genome sequence data I have? Then if the talks back to you and give the follow-up question. Are you interested in additional information, any related information? That like the way our mind works, we can incorporate into the data set as well to get more and more information. And also we can use artificial intelligence for annotation and eventually assembly pipeline, and then all of them needs to be streamlined from the start to the end. Currently in many places that domain is highly assorted, so we can bring all of them in one location from the start to the end. So coming to the last section, and I was told that there are students attending this workshop. So just for them, I want to share some information to all of you. Few years, 30, 40 years back, we had cell phone, which was bulky, and it was used only for one purpose. Now, cell phone became smartphone, and it made our life very easy, and the whole world is at our fingertips. Same goes for the computers. 40, 50 years back, the computer was room full, and then operated by only experts. But now everyone has computer, and even kids can use the computer without any problem. If that is the case for the hardware, think of it like a data science angle. Banking, one example. 30 years back, we used to stand in the queue in the bank to between 8 to 5 or 9 to 5, and then stand in the long queue and get the money. But now, with online banking, the whole bank is at our fingertips, and we can do at any time, 24/7. Sitting on our couch and having a cup of coffee, we can do that. When it comes to genomics, current scenario, as you can see in many places, it's a command line based, and where it is going to go. So that is the question, $1,000,000 question to everyone. So we have to make the data easily accessible to all the scientists so that we should be able to drive the innovations. So in conclusion, regardless of anything what I mentioned throughout the talk, whether it makes sense or something, but at least I want to conclude the talk with the 3 important messages for all of us is, number 1 is data quality for downstream analysis, because everything starts at that data generation. You probably must have heard that phrase, garbage in, garbage out. So make sure to improve the quality of your genomics data, and that will have a lot of that will benefit all of us at the downstream analysis. And the second thing is, it's not only the quality, data integrity. We need to have that data in one place as a repository, which is easily accessible. The example I told you is for GenBank. If GenBank is not there, how our life would be. So that question we have to ask for every organization, whether it's a private or public institute. And not only the data quality and the data integrity and easy access and get the insights faster, right? Of course, there are, in the overall analysis, we have two parts. Majority of them are very simple tasks, like looking for homology analysis, comparative genomics, and so on and so forth. But there are some complex analyses there. But that we will leave it to experts. But at least the most routine work, like variant analysis, right now became almost coming to routine. All of them we can give back to everybody to run the analysis. And in order for us to do that, we need to build user-friendly platforms. So that is meaning, in terms of the skill set development, I know every scientist is going for data science, but along with that, please improve your skill set for user-friendly software platform as well. So in summary, I want to say is we are in a golden era of the genomics. We are generating unprecedented amount of genomics data. We have the opportunity to generate, understand the organism at the holistic level. So let's make the best out of that. So lastly, I want to thank the organizing committee for inviting me to give the keynote address. And also I would like to thank the Carriosoft team and our former and current collaborators and faculties and students who are attending this workshop. So funding, we are supported by both grants and then investment because we are startup, right? So lastly, let's bring the gap between the genomics data and the scientists and bring the data to the fingertips of the scientists and make the scientist happy. Thank you.
Thank you very much, Dr. Rajesh, for giving an excellent overview regarding the marker technology, then genomic data, and how to apply them to develop the new plants for the breeders. So overall, it was a complete story from markers to developing new varieties, and also highlighting the importance of genomic data. So it was very interesting, and I'm sure we will be very keen to know more about your work. So, any questions from the audience?
Can I ask one question? No, it's not
Sure.
a question, it is the comment. Rajesh, I'm in NCL doing chem informatics for so many years. I entered this field, agri, IoT, AI, etc. But today, your talk was an eye opener for me. From plant to plant via data. That means you take the plant, analyze, and then again reintroduce to the plant. Okay, so these are the fantastic things so that we get a better plant in future. That is the theme of your presentation. I am so excited. I am actually, you told the cost reduced so much, $100,000 to $1,000 to $100, and the data growing from GB to terabyte to now eventually to zettabyte, more than astronomical. So now it is a very timely seminar, presentation, lectures. Thanks to organizers to bring people like you to tell that what are the challenges we are going to face and how we are going to use the technology to solve the problems. So with this, actually it's a comment, but I am looking forward to collaborate and learn from all of us so that we can actually deliver something with this knowledge under Bharat Agri-Grid. So this is my comment. So now I have only one simple question. So you told that it takes 6 to 7 generations to get the more than 98% improvement or whatever. Now you told using the technology we can reduce 2 to 3 generations. So can you just tell us in brief? I think you understood what I am talking about. So 6 to generation it takes usually Using the technology, we can reduce 2 to 3 generations. The entire concept of this workshop or conference or Bharat Agri-Grid, can we reduce it 2 to 3 generations? Basically the time. Okay, so if you can tell in that, when some nutshells, it can become the minutes of the meeting to justify. We are going to handle big data. We are going to use very complex algorithms and methods, and we need a huge computing power, and we need infrastructure at the institutional level. So all can be justified with your statement, because we are going to reduce 2 to 3 generations among the 6 to 7 generations. Sorry, I took a big question. Milind, I'm really sorry. I have to give some annotation plus, some questions with the context.
I got the gist of your question, but it's a 5:15. Everybody wants to leave, I think. So let me
No,
be
this is
brief.
very important question, Rajesh.
No, I will give the answer.
Sorry.
See, the simple statistics is when you make one line constant, another one when you make, you get 50% comes from one genome, 50% comes from another genome, correct? That is how progenies are made in general. So the 50, when you are crossing with the one of the original parents, the elite parents, that is going to increase the genome component of the elite parent, reduce the genome amount of the donor line, right? So like that, when you go one by one, so when you see the distribution wise, quantitative, parabolic distribution, our goal is always to select the one that is in this side, which is the highest, the line with the highest percentage of your recurrent parent. But the median would be 50, 75, and 82.5, like that. That is the way the distribution will remain. Imagine when you are making selection without looking into the genome on this end, or the median in the level when you take it. So in order to reach that 80%, it takes several generations, right? Without applying any technology, randomly you are selecting and going through that. But by marker assist selection, you can look at this extreme at every generation, which is at that towards that 80% and 90% like that. So you can select them, so that way you can reduce the time. In the traditional way, it can go up to BC6 to have that close to 98%. This way you can get the 98% at the BC3 level, backcross generation 3 level, at 3 or 4 like that. So that is what basic analysis. That is where the molecular markers play a key role on that. So
excellent, Rajesh. We are going to come back again how to implement in Bharat Agri-Grid, so that for all crops, for the benefit of the society. So I am done with my questions. Thank you so much.
Thank you very much to all of you.
Thank you, Dr. Rajesh, for accepting our invitation and giving your valuable insights in this keynote session. We look forward to have a long collaboration with you.
Certainly. And now my time is going for breakfast while you are going for your dinner. So
Yeah, thanks a lot for accepting invitation.
Thanks a lot, Dr. Aruna. Nice meeting all of you.
Yeah.
Thank you.
Thank you, Rajesh.
Thank you. Yes.
Good day.
Thank you, sir.
So with this, we conclude our today's entire day program. And so tomorrow, look forward to meeting everyone at 10:30.
Yes, yes. Yeah, 10:30
Thank
will
you.
start second day workshop for all the participants, please.
Ma'am, I'm
Thank you
sorry I
everyone
took
for
long time to
joining.
ask questions and clarification. And this
No,
is
it's
very
really
critical
required.
for the proposal.
Sir, there are so many participants on YouTube. They are not visible here.
I see.
It is
Thank you.
YouTube live streaming, so many participants are there.
Excellent.
Because,
Okay, thank you.
registration was filled, na? So participants on YouTube can also put questions. We'll take a few questions from that, please. Rajesh, we can do that, na? Rajesh, are you there? Who is there?
Dr. Aruna, please send us the YouTube link. I will share with some students here.
Okay, Ayan. Okay, sure. We'll share the link.
I will forward the mail.
Yeah, Om Prakash, please. So I already informed to Rajesh, if some questions, then he will send it to us here in the chat box. Okay. Thank you, everyone.
Thank you. Thank you.
Okay.
Thank you, ma'am. Okay.
Okay, bye.
Thanks, Kathy.